ಹಲೋ ಎವ್ರಿವಾನ್, ಎಲ್ಲರಿಗೂ ನಮಸ್ತೆ. ಇವತ್ತು ನೀವು ಇವಾಗ ಮ್ಯಾಥ್ಸ್ ಮತ್ತು ಬಯಾಲಜಿ ಪೇಪರನ್ನು ಬರೆದಿದ್ದೀರಾ CET KCET 2024. ಸೊ ಬಯಾಲಜಿ ಪೇಪರ್ ನಿಮಗೆ ಬೆಳಗ್ಗೆ ಇತ್ತು. ಸೊ ಯು ಹ್ಯಾವ್ ಫಿನಿಶ್ಡ್ ವಿತ್ ದಿ ಬಯಾಲಜಿ ಪೇಪರ್. ಸೊ ಲೆಟ್ ಅಸ್ ಡಿಸ್ಕಸ್ ಒಂದು ಸಾರ್ಟ್. ಈಗ ಲೆಟ್ ಅಸ್ ಡಿಸ್ಕಸ್ ವಿತ್ ಒನ್ ಆಫ್ ದಿ ವರ್ಷನ್. ನಮ್ಮ ಹತ್ರ ಇವಾಗ ನಾನು ವರ್ಷನ್ D ಇದೆ. ಸೊ ವರ್ಷನ್ D ಅನ್ನು ಡಿಸ್ಕಸ್ ಮಾಡಿಕೊಂಡು ಹೋಗೋಣ, ಆಯಿತಾ? ಸೊ ಫಸ್ಟ್ ಕ್ವಶನ್, ವರ್ಷನಲ್ಲಿ ಫಸ್ಟ್ ಕ್ವಶನ್ ಏನಿದೆ ಅಂತ ಹೇಳಿದ್ರೆ ಜಿನೋಟೈಪ್ ರೇಷಿಯೋ ಫಾರ್ ಇನ್ಕಂಪ್ಲೀಟ್ ಡಾಮಿನೆನ್ಸ್ ಅಂತಿದೆ. ಸೊ ನಿಮಗೆ ಗೊತ್ತಿದೆ ಇನ್ಕಂಪ್ಲೀಟ್ ಡಾಮಿನೆನ್ಸ್ ಅಂದ್ರೆ ಏನು ಅಂತ ಹೇಳಿ, ಅಲ್ವಾ? ಸೊ ಇನ್ಕಂಪ್ಲೀಟ್ ಡಾಮಿನೆನ್ಸ್ ಅಂತ ಹೇಳಿದ್ರೆ ಏನು? ಸೊ ನಿಮಗೆ ಎರಡು ಅಲೀಲ್ಸ್ ಅನ್ನ, ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ರೆಡ್ ಮತ್ತೆ ವೈಟ್, ರೆಡ್ ಅಂಡ್ ವೈಟ್ ಫಿನೋಟೈಪ್ಸ್ ಅನ್ನ ಕ್ರಾಸ್ ಮಾಡಿದಾಗ we get hybrid. But hybrid ಏನು ಮಾಡುತ್ತೆ? hybrid will give you pink colorations, right? So intermediate coloration ಸಿಗ್ತದೆ. ಸೊ ಈ intermediate coloration ಗೆ ಯಾವ ಜಿನೋಟೈಪಿಕ್ ರೇಷಿಯೋ ಇದೆ, ಜಿನೋಟೈಪ್ ರೇಷಿಯೋ ಏನಿದೆ, ಯಾವ ಇಂಟರ್ಮೀಡಿಯೇಟ್ ರೇಷಿಯೋ ಇರುತ್ತೆ ಅಂತ. ಸೊ ನೀವು F2 ಜನರೇಷನ್ಗೆ ಹೋದಾಗ, when you go to the F2 generation, right, in case of so capital R small r crossed with capital R small r ಮಾಡಿದಾಗ, ಸೊ ನಮಗೆ ಇಲ್ಲಿ ಬರುವಂತಹ ಗ್ಯಾಮೆಟ್ would be capital R small r ಮತ್ತೆ capital R small r, ಅಲ್ವಾ? ಸೊ we get capital R that is red, capital R small r, capital R small r ವಾಪಸ್, or let us do it in the Punnett square ಗೆ ಹಾಕಿದ್ರೆ, let us do that in a Punnett square. So capital R and a small r. So these are the two gametes. ಎರಡು gametes ಸಿಕ್ಕಿದೆ. ಸೊ ನಾವು ಈ gametes ಅಲ್ಲಿ ನೋಡಿದ್ರೆ, so what do we get? capital R, capital R, small r, then capital R, small r, small r, small r. So ಇಲ್ಲಿ red, pink, then again ಇಲ್ಲಿ ನಿಮಗೆ pink flower ಸಿಗ್ತದೆ, where ಇಲ್ಲಿ ನಿಮಗೆ white flower ಸಿಗ್ತದೆ. So they are in the ratio 1:2:1. Okay, so the correct answer is 1:2:1, right? So 3:1 ಅಂತ ಕೊಟ್ಟಿದ್ದಾರೆ. 3:1 ಯೂಶುವಲಿ ನಿಮಗೆ ಫಿನೋಟೈಪಿಕ್ ರೇಷಿಯೋ. ಯಾವಾಗ ಫಿನೋಟೈಪಿಕ್ ರೇಷಿಯೋ ಬರ್ತದೆ? ಮೊನೋಹೈಬ್ರಿಡ್ ಕ್ರಾಸ್ ಅಲ್ಲಿ. ಮೊನೋಹೈಬ್ರಿಡ್, when we have monohybrid, 3:1 phenotypic ratio, not genotypic ratio, phenotypes, right? Phenotypic ratio ಬರ್ತದೆ. where 9:3:1 ಬರುವಂತದ್ದು ನಿಮಗೆ ಇನ್ಕಂಪ್ಲೀಟ್ ಡಾಮಿನೆನ್ಸ್ ಅಲ್ಲಿ, ರೈಟ್? ಇನ್ಕಂಪ್ಲೀಟ್ ಡಾಮಿನೆನ್ಸ್ ಅಲ್ಲಿ, ಸಾರಿ, 9:3:1 ಬರುವಂತದ್ದು ನಿಮಗೆ ಡೈಹೈಬ್ರಿಡ್ ಕ್ರಾಸ್, ದಟ್ ಈಸ್ ಇಂಡಿಪೆಂಡೆಂಟ್ ಅಸಾರ್ಟ್ಮೆಂಟ್ ಲಾ. ಸೊ ಇಂಡಿಪೆಂಡೆಂಟ್, ನಿಮ್ಮ ಜೀನ್ಸ್ what we have independently assorted, right? Independently assort ಆದಾಗ ಬರ್ತದೆ. So this is your dihybrid cross. Okay, so this would be your dihybrid cross. Okay na? So the first question ಗೆ answer ಏನು? 1:2:1. Okay, so let's move to the next question. Next question ಏನು ಕೊಟ್ಟಿದ್ದಾರೆ ಅಂತ ಹೇಳಿದ್ರೆ, find the incorrect statement among the following. ಇದರಲ್ಲಿ incorrect ಯಾವುದು ಅಂತ ಹೇಳಿದ್ದಾರೆ. So in sex linked recessive trait, the gene is transmitted from unaffected carrier, right? If sex linked recessive trait, gene is connected with the unaffected carrier, female to some of the male progeny ಅಂತ ಕೊಡ್ತಾರೆ. ಸೊ ವೆದರ್ ಇಟ್ ಇಸ್ ಕರೆಕ್ಟ್ ಆರ್ ರಾಂಗ್ ಅಂತ. ಸೊ ನಿಮಗೆ ಇಲ್ಲಿ ಏನಾಗುತ್ತೆ? ಸೆಕ್ಸ್ ಲಿಂಕ್ಡ್ ರಿಸೆಸಿವ್ ಟ್ರೈಟ್ ಅಂತ ಹೇಳಿದರೆ, ಸೆಕ್ಸ್ ಲಿಂಕ್ಡ್ ರಿಸೆಸಿವ್ ಟ್ರೈಟ್ ಅಂದ್ರೆ ಏನು? ಸೊ ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಲೆಟ್ ಅಸ್ ಟೇಕ್ ಯುವರ್ ಕಲರ್ ಬ್ಲೈಂಡ್ನೆಸ್ ತಗೊಳ್ಳೋಣ. ಅದು ಯಾವತ್ತು ರಿಸೆಸಿವ್, ನಿಮಗೆ ಎರಡು ಕ್ರೋಮೋಸೋಮ್ಸ್ ಅಲೀಲ್ಸ್ ಬಂದಾಗ, ಎಕ್ಸ್ ಕ್ರೋಮೋಸೋಮ್ಸ್, ಬೋತ್ ಎಕ್ಸ್ ಕ್ರೋಮೋಸೋಮ್ಸ್ ಅಲ್ಲಿ ನಿಮಗೆ ಸ್ಮಾಲರ್ ರಿಸೆಸಿವ್ ಅಲೀಲ್ಸ್ ಇದ್ದಾಗ ಏನಾಗುತ್ತೆ? they will show, or your characters would be expressed, ಅಲ್ವಾ? ನಿಮ್ಮ ಕ್ಯಾರೆಕ್ಟರ್ ಎಕ್ಸ್ಪ್ರೆಸ್ ಆಗುತ್ತೆ. ಸೊ character would be expressed. So but in case of female ಏನಾಗುತ್ತೆ capital C ಇದ್ದು small c ಇದ್ರೆ ಅಂದ್ರೆ ನಿಮ್ಮ dominant allele ಮತ್ತೆ recessive allele ಇದ್ರೆ she is normal ಅಲ್ವಾ, she is normal but she is carrier. ಇವಾಗ ಇವರು ಅದನ್ನೇ ಕೇಳಿದ್ದಾರೆ. ಸೊ ಇಲ್ಲಿ ನನ್ನ ಹತ್ರ ಸೆಕ್ಸ್ ಲಿಂಕ್ಡ್ ರಿಸೆಸಿವ್ ಟ್ರೈಟ್ ದ ಜೀನ್ ಇಸ್ ಟ್ರಾನ್ಸ್ಮಿಟೆಡ್ ಫ್ರಮ್ ಅನ್ಅಫೆಕ್ಟೆಡ್ ಕ್ಯಾರಿಯರ್ ಅಂತ ಕೇಳಿದ್ದಾರೆ. ಅನ್ಅಫೆಕ್ಟೆಡ್ ಕ್ಯಾರಿಯರ್ ಫೀಮೇಲ್. ಸೊ ವೆನ್ ಅನ್ಅಫೆಕ್ಟೆಡ್ ಕ್ಯಾರಿಯರ್ ಫೀಮೇಲ್ ಅಂತ ಹೇಳಿದ್ರೆ ಕ್ಯಾಪಿಟಲ್ ಸಿ ಕ್ರಾಸ್ ವಿತ್ ಸ್ಮಾಲ್ ಸಿ ವೆನ್ ಶಿ ಮ್ಯಾರಿಡ್ ಟು ಅ ನಾರ್ಮಲ್ ಮೆನ್ ಅಂತ ಹೇಳಿದ್ರೆ, ಸೊ ರಿಮೆಂಬರ್ ದ್ಯಾಟ್ ಸೊ ಶಿ ಮೇ ಹ್ಯಾವ್ normal boy or a affected boy. So the progeny may be affected or normal like this. Okay, so this is affected. This is affected progeny. Illi enagutte he will be normal. This is normal male. This is normal male. Okay, normal or affected male cell. So illi accumulations of phenyl pyruvic acid. So this statement is correct, right? So e statement kuda correct. Accumulations of phenyl pyruvic acid in brain results in the mental retardation. Yes, accumulations of phenyl pyruvic acid yavaga agtade by a specific enzyme, right? What is that enzyme? Specific enzyme is phenylalanine hydroxylase enzyme. Phenylalanine hydroxylase enzymes the mutation ಆದರೆ ನಿಮಗೆ phenyl pyruvic acid accumulation ಆಗ್ತದೆ. As a result phenyl ketonuria formation ಆಗ್ತದೆ, which is responsible for your mental retardation. This is also correct statement. Individuals affected by the Down syndrome will have a congenital heart defect and are more intelligent ಅಂತ ಹೇಳ್ತಾರೆ. So they are not. So Turner syndrome is caused due to the absence of one X chromosomes. Our phenotype would be X naught. It is correct. This is also correct. This is also correct. And the first statement is also correct. So our correct statement, correct answer for this question would be incorrect statement. So individuals affected the Down syndrome will have a congenital heart defect and are more intelligent. ಅನ್ನುವಂಥದ್ದು wrong statement. So the option should be C for the question number 2. Okay, next question ಗೆ ಹೋಗುವ ಆಯ್ತಾ. ಇಲ್ಲಿ ಏನು ಕೇಳಿದ್ದಾರೆ, in a hybrid cross between two breeding round yellow seeded true breeding wrinkle green seeded pea plant the ratio of segregation ಏನಂತ ಕೇಳಿದ್ದಾರೆ. in F2 generation ರೇಷಿಯೋ ಆಫ್ ಸೆಗ್ರಿಗೇಷನ್ ಎಷ್ಟಿರ್ತದೆ ನೆನ್ಪಿಟ್ಕೊಳ್ಳಿ. ಇನ್ ರೇಷಿಯೋ ವುಡ್ ಬಿ 3:1 ಇರ್ತದೆ ಆಯ್ತಾ. ಸೊ ನಿಮಗೆ ರೇಷಿಯೋ ಏನಿರ್ತದೆ, ಇಲ್ಲಿ ಸೆಗ್ರಿಗೇಷನ್ ಕೇಳಿದ್ದಾರೆ. ಸೆಗ್ರಿಗೇಷನ್ ಅರ್ಥ ಏನು, ಈಗ ನಾವು ದೇ ಹ್ಯಾವ್ ಸೆಡ್ ರೌಂಡ್ ಯೆಲ್ಲೋ ಸೀಡ್, ರೌಂಡ್ ಯೆಲ್ಲೋ ಕ್ಯಾಪಿಟಲ್ R capital Y small y crossed with wrinkled green seed. Wrinkled green would be smaller crossed with ಸ್ಮಾಲ್ y ಇರ್ತದೆ. ಸೊ ನಿಮಗೆ ನಾವು ಕ್ರಾಸ್ ಮಾಡಿದಾಗ ಇವೆರಡನ್ನು ಕ್ರಾಸ್ ಮಾಡಿದಾಗ ನಮ್ಗೆ ಏನು ಸಿಗ್ತದೆ, ಕ್ಯಾಪಿಟಲ್ R ಕ್ಯಾಪಿಟಲ್ Y ಸ್ಮಾಲ್ y ಸಿಕ್ತು. ಸೊ ದಿಸ್ ಇಸ್ ಅಗೈನ್ ರೌಂಡ್ ಯೆಲ್ಲೋ. ಫಿನೋಟೈಪ್ ಈಸ್ ರೌಂಡ್ ಯೆಲ್ಲೋ ಅಲ್ವಾ. ಸೊ ಈ ರೌಂಡ್ ಯೆಲ್ಲೋ ಫಿನೋಟೈಪ್ ಯಾವುದು ಇನ್ ಕೇಸ್ ಆಫ್ F1 ಜನರೇಷನ್. ಸೊ ನಾವು ದಿಸ್ F1 ಜನರೇಷನ್ ಫಿನೋಟೈಪ್ ನೀವು F2ಗೆ ಕ್ರಾಸ್ ಮಾಡಿದಾಗ ಇನ್ F2 ಜನರೇಷನ್ ವಾಟ್ ಈಸ್ ದಿ ಸೆಗ್ರಿಗೇಷನ್ ಅಂತ ಹೇಳಿದ್ದಾರೆ. ಸೊ ಸೆಗ್ರಿಗೇಷನ್ ಅಂತ ಹೇಳಿದ್ರೆ ಏನು, ಗ್ಯಾಮಿಟ್ ಫಾರ್ಮೇಷನ್ ಆಗುವಾಗ ಇಂಡಿಪೆಂಡೆಂಟ್ ಅಸಾರ್ಟ್ಮೆಂಟ್ ಲೋ ಆಯ್ತು, ರೈಟ್. ಇಂಡಿಪೆಂಡೆಂಟ್ ಅಸಾರ್ಟ್ ಆದಾಗ ನಿಮ್ಮ ಯೆಲ್ಲೋ ಅಂಡ್ ರೌಂಡ್, ರೈಟ್, wrinkled green ಅಂತ ಉಂಟು ಅಂಡ್ ರೌಂಡ್ ಯೆಲ್ಲೋ wrinkled green ಇದೆ. ಯೆಲ್ಲೋ ಅಂಡ್ ಗ್ರೀನ್ ಸೆಗ್ರಿಗೇಟ್ ಆಗುತ್ತೆ, ಇದು ಮತ್ತೆ ಇದು ಸೆಗ್ರಿಗೇಷನ್ ಆಗುತ್ತೆ. ಎಷ್ಟು 3:1. Okay, so this is in case of wrinkled yellow matte green segregated into 3:1 yellow green. Same way ನಿಮ್ಮ round matte wrinkled kuda segregate agutte, again 3:1. So answer should be 3:1 for this specific question. Okay, so question number 3, answer what we have is 3:1. Okay na. So next illi enagutte, next question. The government of India is legalized MTP with some strict regulations in the year. Yavaga aitu, when the year they have regulated the strict regulations of MTP. What do you mean by MTP? Medical termination of pregnancy. Medical termination of pregnancy. Usually ever in martidro, so they usually after knowing that the sex of the child, if it is female, avaga ಏನು ಮಾಡ್ತೀವಿ, ಮೆಡಿಕಲ್ ಟರ್ಮಿನೇಷನ್ಗೆ ಹೋಗ್ತೀವಿ. ಸೊ ಅವಾಗ ಸ್ಪೆಸಿಫಿಕ್ ರೂಲ್ಸ್ ಅಂಡ್ ಫೀಮೇಲ್ ಫಿಟಿಸೈಡ್ ಜಾಸ್ತಿ ಆಯಿತು ಅಲ್ವಾ, ಫೀಮೇಲ್ feticide is increased. Okay, female feticide is increased. So that is why what they did, they have regulated some rules and regulations so that they can reduce all these illegal activities like female feticides and all. Okay, so that rule has been given in the year or about legalized, MTP would be legalized, regulated in the year 1971. Okay, so the direct question, so we do not have much explanations for that. It's a direct question. So it was in the year 1951. Okay, so let's move on to the next question. The process of in which small part of the vas deferens is cut, right, or removed and tied up through a small incision called ಏನಂತ ಕೇಳಿದ್ದಾರೆ. So process of small part of the vas differences, vasa differentia cut ಮಾಡ್ತಾ ಇದ್ದಾರೆ. This is a sterilization process, right? So this process ಏನಿದೆ, ಇದು sterilization process. So the answer is vasectomy, right? Answer is vasectomy, sterilization process. Where in case of the tubectomy anta helidre, fallopian tube is cut. Tubectomy is also sterilization process where there is an incision. Okay, incision in fallopian tube. Okay, incisions in the fallopian tube, which is also again sterilization process, type of sterilization process in case of females, to restrict both process, to restrict mating of sperm with egg. Okay na. So answer ಏನಾಗುತ್ತೆ ನಿಮ್ದು vasectomy is the answer. So where MTP, we know that medical termination of pregnancy, and gift ಅಂತ ಹೇಳಿದ್ರೆ gamete intrafallopian transfer. Okay, so where the gametes are introduced into fallopian transfer, majorly sperm is introduced into fallopian tube. So this would be your answer, fifth question, that is the if the vas difference is in situ. So the answer is vasectomy. Okay, let's go to the next question. Test cross in pre-plant. We know that. What do you mean by test cross is? Test cross is majorly did or conducted to identify its phenotypic and genotypic ratio. So majorly genotype. What type of genotype is? It is majorly conducted to identify the genotype of a plant for a specific characteristic. ಒಂದು ಸ್ಪೆಸಿಫಿಕ್ ಕ್ಯಾರೆಕ್ಟರಿಸ್ಟಿಕ್ಸ್ಗೆ ಏನು ಜಿನೋಟೈಪ್ ಇದೆ, ಅದರ ಜಿನೋಟೈಪ್ ಏನು ಅಂತ ನೋಡಲಿಕ್ಕೆ. So what do you mean by test cross? Test cross is any progeny, your F1 progeny or F2 progeny. Okay, so ಇಲ್ಲಿ F1 progeny ಅಂತ ಕೊಡ್ಲಿಲ್ಲ ಯಾವುದು ಕೂಡ, ಎಲ್ಲ ಕಡೆ F2 progeny ಅಂತ ಕೊಟ್ಟಿದ್ದಾರೆ, ಎಕ್ಸೆಪ್ಟ್ ಇಲ್ಲಿ F1 ಪ್ಲಾಂಟ್ ಅಂತ ಮಾತ್ರ ಕೊಟ್ಟಿದ್ದಾರೆ. ಬಟ್ ಅವರು ಸ್ಪೆಸಿಫೈ ಮಾಡಲಿಲ್ಲ, ಕ್ರಾಸ್ ಬಿಟ್ವೀನ್ F1 ಅಂತ ಮಾತ್ರ ಮೆನ್ಷನ್ ಮಾಡಿದ್ದಾರೆ. ಬಟ್ ವಾಟ್ ಯು ಮೀನ್ ಬೈ ಟೆಸ್ಟ್ ಕ್ರಾಸ್? ಸಿ ನಿಮಗೆ ಎರಡು ಬರುತ್ತೆ, ಬ್ಯಾಕ್ ಕ್ರಾಸ್ ಬರುತ್ತೆ. ಸೊ ಬ್ಯಾಕ್ ಕ್ರಾಸ್ ಈಸ್ ಕ್ರಾಸಿಂಗ್ ಆಫ್ hybrid with any of the parent, like dominant parent or a recessive parent. So any of the parent, so either this parent or this parent, so this become a back cross. Where test cross anta bandaga, when we speak about test cross anta bandaga, the test cross is majorly when we cross our hybrid with recessive parent. So remember that it is for the recessive parent. So the recessive parent is your F2 generation, a cross of a cross between F2 tall plants with the recessive parent. Okay, tall plant with the recessive parent. Illi dwarf plant with the recessive parent bartide. Dwarf plant with the recessive parent bandre yellow kuda dwarf agtade. When we cross this one and this with the recessive parent, all hybrids are right, all of them are dwarf, right, all of them are dwarf. But where here you can make out that is 1:1 ratio. Okay, so they are in the 1:1 ratio. So option is, answer is test cross in a pea plant is a cross between F2 tall plant with a recessive parent. Okay, so let's move on to the next question. Homeostasis in the condition where the organisms are. What do you mean by homeostasis? And the first now note kodbeku. What do you mean by homeostasis? Homeostasis anta helidre eno, when we speak about homeostasis anta helidre, so we'll be having or maintaining internal environment. Okay, maintaining internal environment. So that is what we call as homeostasis. Okay, so andre maintaining the internal environment anta helidre yavudu irbodu? For example, temperature irbodu, ನಿಮ್ಮ pH irbodu, ನಿಮ್ಮ ವಾಟರ್ ಕಂಡೀಷನ್ಸ್ ಇರಬಹುದು, ಇವೆಲ್ಲವನ್ನು ನಾವೇನ್ ಮಾಡ್ತೇವೆ, ಮೇಂಟೈನ್ ಮಾಡ್ತೇವೆ. ಸೊ ಆ ಮೇಂಟೈನ್ ಮಾಡ್ತೀವಿ. ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ನಾವು ಮ್ಯಾಮಲ್ಸ್, ರೈಟ್? ಸೊ ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಮ್ಯಾಮಲ್ಸ್, ಬರ್ಡ್ಸ್, ಆಲ್ ಆಫ್ ದೆಮ್ ಆರ್, ನಾವೇನ್ ಕರಿತೀವಿ ಅವರಿಗೆ ವಾರ್ಮ್ ಬ್ಲಡೆಡ್ ಆರ್ಗ್ಯಾನಿಸಮ್ ಅಂತ ಕರಿತೀವಿ. ವಾರ್ಮ್ ಬ್ಲಡೆಡ್ organism. So warm blooded organisms anta helidre which can maintain the internal environment, internal body environment. Okay, so illi en kelidare, homeostasis is a condition where the organisms are maintain a constant internal environment in an over changing external environment anta kelidare, right? So the option is A. So A is the correct option for this. So do not maintain the constant environment antuntu alla. So change their internal environment according to their external environment antuntu. These are called conformers, right? So they are poikilothermic organisms, poikilothermic organisms. Okay, so the option of the question number 7 is homeostasis. That would be A. Okay, so next one, the next question, which of the following is not parasitic adaptations? Parasitic adaptations ಏನೈತೆ? Parasitic adaptations ಅಲ್ಲಿ ನಾವು ಏನೈತೆ? So some of the questions parasitic adaptations ಅಂತ ನೋಡಿದ್ರೆ, so what do you mean? Mainly you have a parasite is dependent on other organisms. So parasites ಅಲ್ಲಿ loss of unnecessary sense organs, ಅವುಗಳಿಗೆ ಸೆನ್ಸ್ ಆರ್ಗನ್ಸ್ಗಳ ಅವಶ್ಯಕತೆ ಇರುವುದಿಲ್ಲ, ಬಿಕಾಸ್ ದೇ ಆರ್ ಕಂಪ್ಲೀಟ್ಲಿ ಡಿಪೆಂಡೆಂಟ್ ಆನ್ ಅವುಗಳಿಗೆ ಸೆನ್ಸ್ ಆರ್ಗನ್ ಅವಶ್ಯಕತೆ ಇರುವುದಿಲ್ಲ. ಸೊ ದೇ ಆರ್ ಡಿಪೆಂಡೆಂಟ್ ಆನ್ ದಿ ಹೋಸ್ಟ್ ಆರ್ಗಾನಿಸಮ್ಸ್. ಸೊ ದಿಸ್ ಇಸ್ correct. Loss of unnecessary sense organs. Exactly, they do not have any sense organ. This is the correct option. Correct. This is also correct. Absence of adhesive organs or suckers. Right. So which of the following is not a parasitic adaptations? Absence of adhesive organs or suckers. So this is absence anta mention madidare. No, illi absence alla, presence. So they will be having adhesive organs or the suckers both, so that ಆಗ್ತಾ ಇದೆ. ಸೊ suckers ಇದೆ. Suckers and adhesive organs, they will be helping for the absorption of the nutrients. ಆಯ್ತಾ? So absorption of nutrient molecule. So option is question number 8 ಗೆ option B is the correct answer. Okay, because loss of digestive system, ಅವುಗಳ digestive system ಇರುವುದಿಲ್ಲ. High reproductive capacity, yes, they are having a high reproductive capacity. ನೀವು parasitic adaptations ನೋಡಿದ್ರೆ, parasitic organisms ನೋಡಿದ್ರೆ, ಅವುಗಳು ತುಂಬಾ organisms ಅನ್ನು ಪ್ರೊಡಕ್ಷನ್ ಮಾಡ್ತವೆ. ಮಲ್ಟಿಪ್ಲಿಕೇಶನ್ ರೇಟ್ ವುಡ್ ಬಿ ಹೈಯರ್. ಆಯ್ತಾ? ಸೊ ಡೈಜೆಸ್ಟಿವ್ ಸಿಸ್ಟಮ್ ಲಾಸ್ ಆಗಿರ್ತದೆ, ಅಂದ್ರೆ ಅವುಗಳಿಗೆ ಡೈಜೆಷನ್ ಮಾಡೋ ಅವಶ್ಯಕತೆ ಇರೋದಿಲ್ಲ. ಯಾಕಂದ್ರೆ ನಾವು ಡೈರೆಕ್ಟ್ಲಿ ನಾವೇನ್ ಮಾಡ್ತೇವೆ, ನ್ಯೂಟ್ರಿಯೆಂಟ್ ಅನ್ನ ಅಬ್ಸಾರ್ಬ್ ಮಾಡ್ತೇವೆ. ದೇ ಅಬ್ಸಾರ್ಬ್ಸ್ ದ ನ್ಯೂಟ್ರಿಯೆಂಟ್, ರೈಟ್? ಸೊ ವೆನ್ ದೇ ಆರ್ ಅಬ್ಸಾರ್ಬ್ ದ ನ್ಯೂಟ್ರಿಯೆಂಟ್, we do not want a digestive system. So option B is the answer, for which is not a parasitic adaptations in case of parasites. Okay, let's move on to the next question. DNA polymerase of Thermus aquaticus is exactly. See, this question is from your biotechnology chapter. You know DNA polymerase, which are responsible. What is the function of the DNA polymerase? DNA polymerases. What is the function of the DNA polymerases? DNA polymerase ಏನು ಮಾಡ್ತದೆ? So they will synthesize new DNA strands. They will synthesize a new DNA strand. So now, when they synthesize a new DNA strands, right? So when they synthesize a new DNA strand, we require a DNA polymerase 1, 2, 3, everything. So majorly 3, which are responsible for synthesizing the new DNA strands. But Thermus aquaticus ಅಂತ ಏನಿದೆ, ಅದು ನಿಮ್ಮದು ಥರ್ಮೋಫೈಲ್. ಥರ್ಮೋಫೈಲ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ, ಥರ್ಮೋಫೈಲ್. ಓಕೆ, ಸೊ ಬ್ಯಾಕ್ಟೀರಿಯಾ ನಿಮ್ಮದು ಆರ್ಕಿ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಏನಿದೆ, ದಿಸ್ ಇಸ್ ಥರ್ಮೋ ಎಸ್ಟೋಫೈಲ್ಸ್ ಅಂತ ನಾವು ಏನು ಕಲ್ತಿದ್ದೇವೆ. ಸೊ ಇದು ಥರ್ಮೋಫೈಲ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ, ಥರ್ಮಸ್ aquaticus. So they are thermostable. So means even thermophiles anta naven kehteve, because they can survive in around 100 degrees Celsius. Okay, so they can survive even in 100 degrees Celsius. They will be surviving in 100 degrees Celsius. So now they can survive in 100 degrees Celsius. DNA polymerase enzyme ನಾವು ಯೂಸ್ ಮಾಡ್ತೇವೆ. ಸೊ ಇದನ್ನ ನಾವು ಎಲ್ಲಿ ಯೂಸ್ ಮಾಡ್ತೇವೆ? ನಿಮ್ಮ ಪಾಲಿಮರೇಸ್ PCR ಟೆಕ್ನಿಕಲ್ ಯೂಸ್ ಮಾಡ್ತೇವೆ. What is that? Polymerase chain reaction ಅಲ್ಲಿ ಯೂಸ್ ಮಾಡ್ತೇವೆ. ಸೊ ನಾವು ಪಾಲಿಮರೇಸ್ ಚೈನ್ ರಿಯಾಕ್ಷನ್ಸ್ ಅಲ್ಲಿ ನಾವೇನ್ ಮಾಡ್ತಿದ್ದೇವೆ? ಸೊ 3 ಪ್ರೋಸೆಸ್ ಬರ್ತದೆ ಪಾಲಿಮರೇಸ್ ಚೈನ್ ರಿಯಾಕ್ಷನ್ ಪ್ರಾಸೆಸ್ ಅಲ್ಲಿ. 3 ಡಿಫರೆಂಟ್ ಸ್ಟೆಪ್ ಇದೆ. ಫಸ್ಟ್ ಒನ್ ಈಸ್ denaturation, alva? So first one is denaturation, next one is annealing, ಮತ್ತೆ ಆಮೇಲೆ ಲಾಸ್ಟ್ ಗೆ extension, right? Extension. So this takes place at 90 degrees Celsius or 92 degrees Celsius, which this takes place around 48 degrees Celsius, where extension takes place about around 72 degrees Celsius. So now at 72 degrees Celsius, ನಮ್ಮ ಬಾಡಿಲಿ ಇರುವಂತ ಬ್ಯಾಕ್ಟೀರಿಯಾಗಳು ನಮ್ಮ ಬಾಡಿಲಿ ಇರುವಂತ ಪಾಲಿಮರೇಸ್ ಎನ್ಸೈಮ್ ಏನಿದೆ, ನಮ್ಮ ಪಾಲಿಮರೇಸ್ ಎನ್ಸೈಮ್ ಏನಿದೆ, it does not work. So it get denatured. So we require a specific polymerase which can survive or which can activate even in case of 72 degrees Celsius. So thermostable. So polymerase enzymes are thermostables, which are extracted from thermophiles, that is your Thermus aquaticus. So the correct option is DNA polymerases of Thermus aquaticus is thermostasis. Okay, let's my next question. If DNA bearing gene of resistant to ampicillin transferred into E. coli cells, host cells become transformed into ampicillin resistant cell. What happened where these E. coli are grown on a medium? If a recombinant DNA bearing a gene, okay, with the ampicillin resistance, so they have an ampicillin resistance for resistance to ampicillin. Okay, so now this gene is transferred, host cell become transformed into ampicillin resistant cell. So ನನ್ನ ಹತ್ರ ಪ್ಲಾಸ್ಮಿಡ್ ಉಂಟು, ನಾನು ಈ ಪ್ಲಾಸ್ಮಿಡ್ಗೆ ನಾನು ಏನು ಮಾಡ್ತಾ ಇದ್ದೇನೆ, ಸೊ ಐ ಹ್ಯಾವ್ ಅ ಪ್ಲಾಸ್ಮಿಡ್. ಈ ಪ್ಲಾಸ್ಮಿಡ್ಗೆ ನಾನು ಏನು ಮಾಡ್ತಾ ಇದ್ದೇನೆ, ಐ ಆಮ್ ಗೋಯಿಂಗ್ ಟು recombinant. I am doing the recombinant. So if a recombinant DNA bearing a gene for resistance to ampicillin, ampicillin resistance ಗೆ gene ನಾನು insert ಮಾಡಿದ್ದೇನೆ. So this become ampicillin resistant gene is inserted to this plasmid. Okay, I don't know what plasmid is. Let us take as a plasmid. That's all. Okay. So now this is inserted into a bacteria, E. coli bacteria. So I have a E. coli bacteria and I have inserted the plasmid consisting of ampicillin resistant gene. So now the host cell becomes transformed into the ampicillin resistant cell. What happens where these E. coli are grown on medium containing ampicillin? So ಈ hosts ಕೂಡ ಏನಾಯಿತು, ಇವಾಗ resistant ಆಯಿತು ಅಲ್ವಾ. So they are also resistant. So now ಇದನ್ನ ನಾನು ampicillin ಇರುವಂತ ಮೀಡಿಯಾಕ್ಕೆ ಹಾಕಿದ್ರೆ, ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಥಿಂಕ್ ದಟ್ ಐ ಹ್ಯಾವ್ ಸೋ ಮೆನಿ ಬ್ಯಾಕ್ಟೀರಿಯಾಸ್, ಸೊ ಈ ಬ್ಯಾಕ್ಟೀರಿಯಾದಲ್ಲಿ ಸೊ ನಮ್ಮ ಹತ್ರ ಏನುಂಟು, ದಿಸ್ ಇಸ್ ampicillin ಆಂಟಿಬಯೋಟಿಕ್. ಓಕೆ, ಸೊ ampicillin ಆಂಟಿಬಯೋಟಿಕ್ ಗೆ ನಾನು ಹಾಕಿದೆ. ಸೊ ampicillin ಆಂಟಿಬಯೋಟಿಕ್ ಗೆ ನಾನು ಈ ಬ್ಯಾಕ್ಟೀರಿಯಾನ ಹಾಕಿದಾಗ what happens ಅಂತ ಕೇಳ್ತಾರೆ. So non-transformant will grow and transformant will die. For example, think that ನಂತರ 2 ಬ್ಯಾಕ್ಟೀರಿಯಾ ಉಂಟು ಅಥವಾ ಇಷ್ಟು ಬ್ಯಾಕ್ಟೀರಿಯಾಗಳಿದ್ದಾವೆ. ಈ ಬ್ಯಾಕ್ಟೀರಿಯಾ non-transformant, this is non-transformant, so this is transformants, right? This is transformants and this is non-transformants. So ಈಗ non-transformants ಇದ್ದಾವೆ, transformants ಇದ್ದಾವೆ. ಸೊ ಇಲ್ಲಿ ಏನಾಗ್ತದೆ ಹಾಕಿದಾಗ, so the answer should be the non-transformants will die and transformants will grow, because non-transformants will die. So those bacteria which died in the those bacteria which are died in this, for example here, okay, so all the bacteria died. So those bacteria are non-transformants, where ampicillin resistant gene iruvanta bacteriaಗಳು survive ಆಗ್ತವೆ, they will grow. So option B is the answer for this question. Non-transformants will die and transformants will grow. Non-transformant ಅಲ್ಲಿ ನಿಮಗೆ ampicillin resistant gene ಇಲ್ಲ. ಸೊ ಆದ್ರಿಂದ ಏನಾಗುತ್ತೆ, ಇವುಗಳು ಇಲ್ಲ, ರೆಸಿಸ್ಟೆಂಟ್ ಜೀನ್ ಇಲ್ಲ, ಈ ಬ್ಯಾಕ್ಟೀರಿಯಾಗಳು ಸತ್ತು ಹೋಗ್ತವೆ, they will die because of the ampicillin antibiotic, where the other will survive. Okay, so the option is B, non-transformants will die and transformants will grow. Okay, next one, question number 11. Which of the following is based upon the principles of antigen antibody infections or interactions? So it's a direct questions, as you know always that we'll be having ELISA. Okay, so ELISA is the answer, enzyme linked immune sorbent assay, where PCR is a multiplication method, rDNA technology, PCR is a multiplication method, where rDNA technology, what we do in case of rDNA technology, we will be inserting or creating a new DNA method or DNA strands, right? So where gel electrophoresis is to separation, gel electrophoresis is separation, of DNA fragments. PCR to amplify amplification of DNA. RDNA technology, where we are developing a new gene, so developing a recombinant DNA. In DNA will changing, we are recombinating. Recombination of gene of interest. Okay, recombination of gene of interest, where ELISA, enzyme linked immunosorbent assay, which is used majorly in case of AIDS and all. Okay, antigen antibody interaction. Based on the antigen antibody interactions, we are going to find out whether our body, ನಮ್ಮ ಬಾಡಿ ಒಳಗಡೆ ಏನಾದರೂ ಒಂದು ಆಂಟಿಬಾಡಿ ಪ್ರೊಡಕ್ಷನ್ ಆಗಿದೆಯಾ, ಆ ಆಂಟಿಬಾಡಿ ಯಾವುದಕ್ಕೆ ಅಂತ ನಾವು ಕಂಡುಹಿಡಿತೇವೆ, then we are going to identify it. So that is what we do in case of your cancers or AIDS test ಮಾಡಿ ಆಯ್ತಾ. Let's move to the next question. It's a very simple question. Among the human health and diseases in the purpose, right, which of the among the following is used to, okay, from the biotechnology application in the, which of the among following is used to treat emphysema anta kodidare. You know what I mean by emphysema is. Emphysema is a respiratory disorder. Emphysema is a respiratory disorder. Okay, so which of the among following used to treat emphysema? So this is from a transgenic animal, right? So in case of this one, transgenic animal, human protein alpha antitrypsin. Okay, alpha antitrypsin, which is used to treat your emphysema. Okay, so human protein is an alpha antitrypsin which are used to treat your emphysema. Lactalbumin, where it will be a malnourished, if a mother is having a less or decreased secretion of milk from mammary gland, then it will be used for the nutrition to the young baby. Okay, so we'll go to the next question. Question number 13. The natural reservoir of the phosphorus is phosphorus. See, the phosphorus and sulfur ಎರಡಿದೆ, right? ನಿಮ್ಮ ಫಾಸ್ಫರಸ್ ಮತ್ತೆ ಸಲ್ಫರ್ ಅನ್ನ ನಾವು ಸೆಡಿಮೆಂಟರಿ ಸೈಕಲ್ ಅಂತ ಕರಿತೀವಿ. ಫಾಸ್ಫರಸ್ ಸೈಕಲ್ and sulfur cycle ಅನ್ನ ನಾವು sedimentary cycles ಅಂತ ಕರಿತೀವಿ. Sedimentary cycle. See, there are different cycles, gaseous cycle and sedimentary cycle. Phosphorus and sulfur are the sedimentary cycles. So remaining carbon dioxide, oxygen, all those things comes under your gaseous cycles. So ಇದನ್ನ ನಾವು ರಿಸರ್ವಾಯರ್ ಎಲ್ಲಿದೆ, ಫಸ್ಟ್ ಎಲ್ಲಿಂದ ಬರುತ್ತೆ? ರಿಸರ್ವಾಯರ್ ಈಸ್ ಇನ್ ಯುವರ್ ರಾಕ್ಸ್. So rocks is the oxygen. Okay, large amount of sulfur and phosphorus are present in case of the sedimentary rocks. So that is why they are called them as a sedimentary cycle. So they are present in the sedimentary rock. Sedimentary rock ಅಲ್ಲಿ ಇರ್ತದೆ. So ಅದರಿಂದ ವೆದರಿಂಗ್ ಪ್ರಾಸೆಸ್ ಆಗ್ತದೆ. ಕೆಮಿಕಲ್ ವೆದರಿಂಗ್ ಇರ್ಬೋದು ಆರ್ ಫಿಸಿಕಲ್ ವೆದರಿಂಗ್ ಇರ್ಬೋದು. ರಾಕ್ ಪುಡಿ ಆಗ್ತದೆ, ರಾಕ್ ಪುಡಿಯಾಗಿ ಮಣ್ಣು ಆಗ್ತದೆ. ಸೊ ಆ ಮಣ್ಣಲ್ಲಿ ನಿಮಗೆ ಫಾಸ್ಫರಸ್ ಬರ್ತದೆ. ಸೊ ನಾವು ಆ ಫಾಸ್ಫರಸ್ ಅನ್ನ ಪ್ಲಾಂಟ್, ಫಾರ್ ಎಕ್ಸಾಂಪಲ್, ವಿಲ್ ಬಿ ಹ್ಯಾವಿಂಗ್ ಇಟ್ ಅ ರಾಕ್. ಓಕೆ, ಸೊ ನಾವು ದಿಸ್ ಫಾಸ್ಫರಸ್ ಈಸ್ ರಿಲೀಸ್ಡ್ ಟು ಯುವರ್ ಸಾಯಿಲ್. Phosphorus is released to soil, and now this phosphorus is absorbed by plants as a nutrient. Okay, nutrient uptake. So now from plant it is transferred to animals. Okay, animal through your food chain, and from animal when animal dies or plant dies, it goes to detritus. Okay, so when plant dies or animal dies, it goes to detritus, and from detritus decomposition ಆಗ್ತದೆ. By decomposition what happen? The organic material breaks down to an inorganic molecule. Okay, so now a part of phosphorus is also released into your water bodies, okay, which is then sedimented So now as a sedimented, this phosphorus, okay, so this amount of phosphorus, the phosphorus is not available for the cycle. So that is why the phosphorus cycle is also called incomplete cycle. Okay, it is also called incomplete cycle. So the reservoir for your phosphorus would be your rock. So option is A for question number 13. Okay, next question. The sequence of communities of the primary succession in water is successions. You know what do you mean by succession is? Succession, you know, sequential change of community is called succession. Okay, it's called succession. So now ಈ ಕೊಟ್ಟದ್ರಲ್ಲಿ ಯಾವುದು ಸಕ್ಸೆಷನ್ ಗೆ ಹೆಲ್ಪ್ ಆಗ್ತದೆ? ಯಾವ ಸಕ್ಸೆಷನ್ ಅಂತ ಕೇಳಿದರೆ ವಾಟರ್ ಸಕ್ಸೆಷನ್ ಅಂತ ಕೇಳಿದರೆ. ಮೀನ್ಸ್ ವಾಟರ್, there are two different types of succession, primary succession and secondary succession. primary succession ಅಂತ ಹೇಳಿದ್ರೆ ಏನು? primary succession ಅನ್ನುವಂಥದ್ದು ನಿಮ್ಮ ಹತ್ರ barren land ಇದೆ, from barren land ಇಂದ it will produces the climax community, then it is primary succession. secondary succession ಅಂತ ಹೇಳಿದ್ರೆ already we have a land that is disturbed, and our disturbed land or disturbed part of the environment or ecosystem develops into a climax community, then it is called as secondary succession. ಇಲ್ಲಿ primary succession ಅಂತ ಹೇಳಿದ್ರೆ ನಮ್ಮ ಹತ್ರ ಈಗ ವಾಟರ್ ಇದ್ದದ್ದು ಡ್ರೈ ಅಪ್ ಆಗಿ ಕಂಪ್ಲೀಟ್ ಫಾರೆಸ್ಟ್ ಫಾರ್ಮೇಷನ್ ಆಗಬೇಕು. ಸೊ ವಿಚ್ ಈಸ್ ದಿ ಆನ್ಸರ್ ಫಾರ್ ದ್ಯಾಟ್ ಅಂತ. ಸೊ ಫೈಟೋಪ್ಲಾಂಕ್ಟಾನ್, ಸ್ಕರ್ಬ್ಸ್, ಫ್ರೀ ಫ್ಲೋಟಿಂಗ್, ರೂಟೆಡ್, ಹೈಡ್ರೋಫೈಟ್ಸ್ ಅಂಡ್ ಗ್ರಾಸ್ ಅಂಡ್ ಟ್ರೀಸ್ ಅಂತ ಉಂಟು. ಸೊ ಫಸ್ಟಿಗೆ ಏನು ಬರಬೇಕು? ಫೈಟೋಪ್ಲಾಂಕ್ಟಾನ್ಸ್ ಬರಬೇಕು. ಎಕ್ಸಾಕ್ಟ್ಲಿ ಫಸ್ಟ್ಗೆ ಫೈಟೋಪ್ಲಾಂಕ್ಟಾನ್ಸ್ ಬರಬೇಕು, ಫಾಲೋಡ್ ಬೈ ರೂಟೆಡ್ ಹೈಡ್ರೋಫೈಟ್ಸ್ ವಿಚ್ ಇಸ್ ಆಕ್ಚುಲಿ ಸಬ್ಮರ್ಜ್ಡ್. ಓಕೆ, ಇದು ಸಬ್ಮರ್ಜ್ಡ್ ರೂಟೆಡ್ ಹೈಡ್ರೋಫೈಟ್ಸ್ ಇರ್ತದೆ. ದೆನ್ ಫ್ರೀ ಫ್ಲೋಟಿಂಗ್ ಹೈಡ್ರೋಫೈಟ್ಸ್ ಬರ್ತದೆ. ಸೊ ಒಂದು community ಆದ ಮೇಲೆ will be having the next community. First this is the community, followed by the community is this one, this is the next community which is following now. So floating hydrophytes ಆದ ಮೇಲೆ this community is replaced by reed swamps. Reed swamps is replaced by the marsh meadows. Marsh meadows ಅಂತ ಬರುವಾಗ ಏನಾಗುತ್ತೆ? You will be having less amount of water here. Water condition is completely decreased. ಆಯ್ತಾ? So marshy area ಆಗಿರ್ತದೆ, be having only a small amount of water. So marshy land, so marsh meadows ಬರ್ತದೆ. Marsh meadows then replaced by the scrubs, then scrubs are replaced by the larger trees. So remember that this is your trees, what we have a specific tree ಬರ್ತದೆ. So that becomes your climax community, where phytoplanktons is a pioneer species, first species. Okay, so the sequence of communities of the succession in water is phytoplanktons. Okay, so option D is the answer. Okay, now so next let's move to the next question. Question number 14. So match the following adaptations given in the list with the examples given in the list 2. They select the option showing the correct option. So they list 1 ಕೊಟ್ಟಿದ್ದಾರೆ, adaptations ಕೊಟ್ಟಿದ್ದಾರೆ ಮತ್ತು example ಕೊಟ್ಟಿದ್ದಾರೆ. Biochemical adaptations are usually seen in case of deep sea fishes. The deep sea fishes ಅಲ್ಲಿ biological adaptations ನೋಡಲಿಕ್ಕೆ ಸಿಗ್ತದೆ. So behavioral adaptations were seen in case of your desert behavioral, right? So they'll be having a specific behavior. Desert system ಗೆ higher temperature ಗೆ ನಿಮಗೆ adapt ಆಗಬೇಕು, ಅದಕ್ಕೆ ನಿಮಗೆ ಡೆಸರ್ಟ್ ಇಕೋಸಿಸ್ಟಮ್ಸ್ ಅಲ್ಲಿ ಇರೋದು ಬಿಹೇವಿಯರ್ ಅಡಾಪ್ಟೇಷನ್ಸ್ ತೋರಿಸ್ತವೆ. ಫಿಸಿಯಾಲಜಿಕಲ್ ಅಡಾಪ್ಟೇಷನ್ಸ್ ಅಂತ ಹೇಳಿದ್ರೆ ಏನು, ದೇ ವಿಲ್ ಶೋ ಯು ಸ್ಪೆಸಿಫಿಕ್ ಅಡಾಪ್ಟೇಷನ್ ಸಚ್ ಆಸ್ ಕಂಗಾರು ಕ್ಯಾಟ್. ಓಕೆ, ಕಂಗಾರು ರ್ಯಾಟ್. ಸೊ ಕಂಗಾರು ರ್ಯಾಟ್ ಏನ್ ಮಾಡ್ತದೆ, ಸೊ ದೀಸ್ ಕಂಗಾರು ರ್ಯಾಟ್ಸ್ ವಿಲ್ excrete, kangaroo rat will excrete concentrated urine so that it can store the water molecule, so that it can loss of water molecule na stop ಮಾಡಬಹುದು. Okay, reabsorption of the water ಆಗುತ್ತೆ. So concentrated urine would be excreted. That is in case of the physiological adaptations. Okay, so then morphological adaptations, that is usually in case of your opentia. Okay, morphological adaptations where you have seen opentia ಏನಾಗಿದೆ, leaf is modified into spines, spine formation ಆಗಿದೆ. So leaves are modified into spines. So option is adaptations. What we have, your biochemical adaptation is your deep sea fishes. So option C is the answer. Okay, so first one is Q, that is deep sea fishes. So the option is your C for this match the following question. Next question number 16. The annual net primary productivity of the biosphere is approximately, biosphere anta bandre it will be higher, a big, right, large one. So nimma net primary productivity means what? Net primary productivity is equal to gross primary productivity minus respiration anta barte. Net primary productivity anta helidre gross primary productivity plus respiration. So now ನಿಮ್ಮ ರೆಸ್ಪಿರೇಷನ್ ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, ನೆಟ್ ಪ್ರೈಮರಿ ಪ್ರೊಡಕ್ಟಿವಿಟಿ ಬಂದು, ಓಕೆ, ಸೊ ಅಬೌಟ್ 170 billion tons. Okay, 170 billion tons would be produced total dry weight. So this is respect to dry weight. Okay, so this is equal to dry weight, the total organic matter, dry weight organic matter would be taken into consideration. Organic matter. Okay, biosphere reserve is a large area. Okay, biosphere reserve would be the large area. So annual net productivity would be 170 billion tons. Okay, so all these are the smaller one. If you consider 55 million tons, what we have. Okay, the productivity of the ocean. If you consider the productivity of ocean ತಕೊಂಡ್ರೆ ನಿಮ್ಮದು 55 billion tons ಬರ್ತದೆ. ಎಲ್ಲಿ? ನಿಮ್ಮ ಓಷನ್ ಅಲ್ಲಿ ನೆನ್ಪಿಟ್ಕೊಳ್ಳಿ. ಓಕೆ, ಓಷನ್ ಗೆ ಬರುವಾಗ ಅದು ನಿಮ್ಮ 70% ಗೆ ಬರ್ತದೆ. ನಿಮ್ಮ ಯಾವುದು ಲ್ಯಾಂಡ್ ಮಾಸ್ ಅಲ್ಲಿ ಆಗುವಂತ ಪ್ರೊಡಕ್ಟಿವಿಟಿಯ 70% would be this one. So in case of 55 million tons, that is in case of ocean. Okay, so the option for the question number 16 would be 170 billion tons. Okay, next question. The strict protection of biodiversity hotspots could reduce the ongoing mass extinction by almost. See, now biodiversity hotspot anta helideve. Biodiversity hotspot is majorly used or specified based on two criteria. That is, one is endemic species. How many endemic species you have? At the same time, what we have, endemic species, and also the area what we have threatening. Okay, so endemic species and the species we have, endemic species and threatened species we have. What do you mean by endemic species? Endemic species anta helidre restricted to to a specific area and not found anywhere else. Okay, restricted to a specific area and not found anywhere else. It is called endemic species. And threatened species, number of threatened species and number of endemic species forms your biodiversity hotspot. So now in India we have 2 hotspots. India, there are 2 biodiversity hotspot. Okay, in India there are 2 biodiversity hotspots. So one is Western Ghats region, ಒಂದು ವೆಸ್ಟರ್ನ್ ಘಾಟ್ ರೀಜನ್ ಬರ್ತದೆ, ಮತ್ತೊಂದು ಹಿಮಾಲಯನ್ ಫುಟ್ ಹಿಲ್ಸ್. foothills of Himalaya or Himalayan foothills. So this will majorly to control the extinction of the species from the wild. Okay, so about 30% of the species, answer is 30% of the species reduced, okay, to get extincted. Got it? So the option of the 17th question, the strict protection of the biodiversity hotspots could reduce the ongoing mass extinction by almost 30%. So there are 6 mass extinction ಆಗ್ತಾ ಇದೆ. This would be the 6th mass extinction. See, they mentioned about the ongoing mass extinction. They mentioned about that. There are already, the world has already faced 5 mass extinction. They have already faced the 5 mass extinction, and now we are facing the 6th one, that is due to anthropogenic activities. Due to anthropogenic activities. So 6th mass extinction 30% adana reduce ಮಾಡುತ್ತೆ. So question number 17, answer would be C, 30. Okay, we'll move to the next question. A direct question. Identify the incorrect match with respect to the recently extinct animals of their place of extinction from the red list. So ಎಲ್ಲೂ ಕೂಡ ಮ್ಯಾಚ್ ಆಗಿದೆ. ಡೋಡೋ ಮಾರಿಷಿಯಸ್, ಕ್ವಾಗಾ ಆಫ್ರಿಕಾ, ಥ್ಯಾಲಿಸೈನ್ ಆಸ್ಟ್ರೇಲಿಯಾ, ಬಟ್ ಸೆಲ್ಲಾ ಸ್ಟೀಕವ್ ಅಂತ ಕೊಟ್ಟಿದ್ದಾರೆ, ವಿಚ್ ಇಸ್ ನಾರ್ತ್ ಅಮೇರಿಕಾ ಅಂತ ಉಂಟು. ಇದು ನಾರ್ತ್ ಅಮೇರಿಕಾ ಅಲ್ಲ, ಇದು ನಿಮಗೆ ರಷ್ಯಾ ಆಗಬೇಕು. ಸೊ ಆಪ್ಷನ್ ಡಿ is the answer. Okay, so question number 19. According to hypothesis proposed by environmental biologists, a relatively constant environment in tropic promotes. In tropic regionally, will be having higher biodiversity, with respect to the temperate zones. Okay, tropic region we have tropics have more biodiversity compared to temperate zones. So according to hypothesis proposed by environmental biologists, a relatively constant environment in tropic promotes. Again promote ಮಾಡ್ತಿದೆ, as I already mentioned you, higher biodiversity ಅಂತ ಮೆನ್ಷನ್ ಮಾಡಿದೆ ನಾನು ಅಲ್ವಾ. ಸೊ ಅಬ್ವಿಯಸ್ಲಿ ನೀವು ಫೈಂಡ್ ಔಟ್ ಮಾಡಬೇಕು ಗ್ರೇಟರ್ ಡೈವರ್ಸಿಟಿ ಎಲ್ಲಿ ಉಂಟು ಗ್ರೇಟರ್ ಸ್ಪೀಶೀಸ್ ಸ್ಪೆಷಲೈಸೇಷನ್ ಅಂಡ್ ಗ್ರೇಟರ್ ಸ್ಪೀಶೀಸ್ ಡೈವರ್ಸಿಟಿ ಅಂತ ಉಂಟು. ಸೊ ಅಬ್ವಿಯಸ್ಲಿ ಸ್ಪೀಶೀಸ್ ಡೈವರ್ಸಿಟಿ is most important. So option B should be the answer. Okay, niche specialization. What do you mean by niche? This should be the answer. Question number 19, B is the answer. Okay, niche ಅಂತ ಹೇಳಿದ್ರೆ ಏನು? Niche ಅಂತ ಹೇಳಿದ್ರೆ, so if you consider niche, if an organism is surviving in a specific area, right? So think that organism X surviving in this area A. area A. Then what is its resources that occupation within an ecosystem? ಈ ಒಂದು ಇಕೋಸಿಸ್ಟಮ್ ಒಳಗಡೆ ಅದು ಯಾವ ಯಾವ ರಿಸೋರ್ಸ್ ಅನ್ನ ಯುಟಿಲೈಸ್ ಮಾಡ್ತದೆ, ಎಲ್ಲಿ ಯುಟಿಲೈಸ್ ಮಾಡುತ್ತೆ, ಸೊ ಅದೆಲ್ಲವೂ ಅದರ ನಿಶ್ ಆಗ್ತದೆ. ಸೊ ಈ ಏರಿಯಾ ಎ ಅಲ್ಲಿ ಈ ಸ್ಪೀಶೀಸ್ ಎಕ್ಸ್ ನ ನಿಶ್ ಏನು, ಇದಿರಬಹುದು ದಿಸ್ ಪಾರ್ಟ್ ಆಫ್ ದಿ ಏರಿಯಾ, this part of the area, and this part of the area, and this part of the area. So ಇದಿಷ್ಟು ಅದರ ನಿಶ್. It goes here, goes here, goes here. ಎಲ್ಲದಕ್ಕೂ ಕೂಡ ಅದರ ಕಂಪ್ಲೀಟ್ ಲೈಫ್ ಸೈಕಲ್ ಎಲ್ಲಿ ಕಂಪ್ಲೀಟ್ ಮಾಡ್ತದೆ, ಅದಕ್ಕೆ ಫುಡ್ ಇರ್ಬೋದು, ಅದರ ಹ್ಯಾಬಿಟ್ಯಾಟ್ ಇರ್ಬೋದು, ಅದರ ಹ್ಯಾಬಿಟ್ ಇರ್ಬೋದು, ಅದರ ರಿಪ್ರೊಡಕ್ಟಿವ್ ಸೈಕಲ್ಸ್ ಇರ್ಬೋದು, ಅದು ಎಲ್ಲಿ ರಿಪ್ರೊಡಕ್ಷನ್ ಮಾಡ್ತದೆ, ಸ್ಪಾನಿಂಗ್ ಇರ್ಬೋದು, ಎನಿಥಿಂಗ್ ಇರ್ಬೋದು, ಫರ್ಟಿಲೈಸೇಷನ್ ಆಗೋದಿರ್ಬೋದು. ಸೊ ಎವ್ರಿಥಿಂಗ್, the whole resources is considered. So what is the resource that used, okay, what is the resource the organism used for the survival? Remember that in a given ecological system, all the resources that is utilized, okay, resource utilized resource utilized by the organism in an ecosystem is called as niche. So ಪ್ರತಿಯೊಂದು organisms ಅಲ್ಲಿ niche specialization ಇದೆ. They want a specific niche, at the same time higher diversity. So the option B is the answer for your question number 19. Okay, next question. In the prevention of air pollution, the role of scrubber is removed. So it is majorly sulfur dioxide. So option C is the answer. Sulfur dioxide is found in case of gaseous stage. So option C is the answer for question number 20. Match the list 1 with the list 2. So this is actually from the environmental issues chapter. So they have given the number of the 4 pollutants and what is their effect. So nitrogen rich fertilizers, where do you see nitrogen rich fertilizers? Okay, that is majorly seen in case of eutrophication. Okay, seen in case of eutrophication. So what do you mean by eutrophication? Usually eutrophication is nothing but your natural death of lake or water body, natural death of lake or water body. So that is rich due to the nutrient enrichment, due to the natural due to nutrient enrichment. Okay, nutrient ಗಳು ಜಾಸ್ತಿ ಆದಾಗ ನೈಟ್ರೋಜನ್ NPK, okay, nutrient is NPK, nitrogen, phosphorus and potassium ಜಾಸ್ತಿ ಆದಾಗ ಏನಾಗುತ್ತೆ, ಅಲ್ಲಿ ಇರುವಂತಹ ವಾಟರ್ ಬಾಡಿಲಿ ಇರುವಂತಹ ಪ್ಲಾಂಟ್ ಸಿಸ್ಟಮ್ಸ್ ಗಳು ಗ್ರೋ ಆಗ್ಲಿಕ್ಕೆ ಶುರು ಆಗ್ತದೆ. As a result, that lake will die. So that is so called as eutrophication. Carbon dioxide is major, right? So carbon dioxide, what we have, which is responsible for greenhouse effect. Remember that, you know what I mean by greenhouse effect is. So which are responsible for the increase in temperature. Okay, greenhouse effect is seen in case of increase in temperature. So that is carbon dioxide is responsible for that. So now what we have, we'll be having carbon dioxide layer in the atmosphere. So now this is your earth will reflects your infrared radiation. So now this infrared radiations, what is reflected is again reflected back into the earth. Okay, the refracted the radiations which is emitted by the earth is reflected back to the atmosphere, again the lower atmosphere. As a result, your temperature increases. So this is what we call as global warming. Okay, this is what we call as global warming, which is caused due to greenhouse effect. So carbon monoxide is a air pollutant, and CFCs which are depleting your ozone. Okay, so which will deplete your ozone O3 into oxygen atom and oxygen. Again, continuous process where we can see the ozone holes. The ozone depletion causing your ozone hole in case of polar region. So option should be B, that is QRSP. Okay, next, move on to the next question. Identify the bacterial disease among the following: brown rust, wheat, tobacco mosaic virus, black rot of crucifers and late blight of potato anta kodidare. So if you consider brown rust wheat anta ide is caused by Pasinia species. Idu Pasinia triticina species in the Pasinia triticina species in the cause agate, which is fungi. Aita? Tobacco mosaic virus, we already know that this is TMV virus, right? Tobacco mosaic virus disease, tobacco mosaic disease is given. Black rot of crucifer anta kodidare. This is usually seen in case of crucifers. Okay, crucifer. Okay, so black rot disease. Black rot disease is caused by a bacteria called Xanthomonas. Bacteria called Xanthomonas campestris. Xanthomonas campestris. So we know that, as I already know that, I have told you that it is bacteria. ಇದಕ್ಕೆ ನಾವು bacterial disease ಅಂತ cause ಆಗುತ್ತೆ. So Xanthomonas campestris ನಿಮ್ಮ bacteria black rot disease ಅನ್ನು cause ಮಾಡುತ್ತೆ. So option ಯಾವುದು? C is the correct option. Late blight potato ಅಂತ ಕೊಟ್ಟಿದ್ದಾರೆ. Late blight potato is again fungi disease caused by Phytophthora species. Phytophthora infestans. Phytophthora infestans species in the late blight potato would be caused. Okay, so idu kuda puna fungi. This is virus. Idu fungi again. So option is C, that is black rot of crucifer. Next question. Now we have a match the following issue. So match the nutrient given in the list 1 with the source list 2 anta kodidare. So idu nimma strategy steps in the bandide. So where the vitamin A, vitamin A enide. So majorly we can see carrots. We already know that we have large carrots are the large source of vitamin A, right? Single cell protein spirulina, right? Single cell protein is spirulina is used in production of the single celled SCP, single celled protein production ಮಾಡಲಿಕ್ಕೆ help ಮಾಡ್ತದೆ. So bitter gourd anta ಕೊಟ್ಟಿದ್ದಾರೆ. Bitter gourd ಅಲ್ಲಿ will be having vitamin C, a large amount of vitamin C in bitter gourd, where in case of beans we'll be having proteins, right? Pulses, so we'll be having protein in case of this one. So with respect to question number 23, option is B is the option, that is R, S, P and Q. Okay, so option B is the correct answer. So next question. The chemical substances which are produced by some microbes which can kill or retarded both of the other microbes are known as. ಎರಡು ಮೈಕ್ರೋಬ್ ಎಲ್ಲವನ್ನು ಕಿಲ್ ಮಾಡ್ತವೆ ಅಂತ ಹೇಳಿ ಆದ್ರೆ ಸೊ ಅಂತ ಇದು ಯಾವುದು ಅಂತ ಕೇಳ್ತದೆ. ಸೊ ದಟ್ ಈಸ್ ಯುವರ್ ಸಿ. ಸ್ಟಾಟಿನ್ ಏನಂತ ನಿಮಗೆ ಗೊತ್ತಿದೆ. ಸ್ಟಾಟಿನ್ ಈಸ್ ಯೂಸ್ಡ್ ಆಸ್ ಕೊಲೆಸ್ಟ್ರಾಲ್ ಲೋವರಿಂಗ್ ಏಜೆಂಟ್, ರೈಟ್? ಕೊಲೆಸ್ಟರಾಲ್ ಲೋವರ್ ಮಾಡ್ತದೆ. ಸ್ಟೆಪ್ಟೋಕೈನೆಸ್ acts as clot busters. Cyclosporins acts as immune suppressive. Immune suppressive, where antibiotic will not allow other organisms to produce, okay, survive. For example, if you are giving some antibiotic, so antibiotically bacteria cannot survive. So this antibiotics either they are extracted illinda fungus inda illandre bacteria extract ಮಾಡ್ತಾರೆ. ಸೊ ಇದು ಒಂದು ಪ್ರೊಡಕ್ಷನ್ ಮಾಡಿರೋದು ಸಬ್ಸ್ಟೆನ್ಸ್, ಕೆಮಿಕಲ್ ಸಬ್ಸ್ಟೆನ್ಸ್ ಎಲ್ಲಿಂದ? ಬ್ಯಾಕ್ಟೀರಿಯಾ ಫಂಗೈ ಇಂದ, ಸೊ ಮೈಕ್ರೋಬ್ ಇಂದ. ಅದೇನ್ ಮಾಡುತ್ತೆ? ಮತ್ತೊಂದು ಫಂಗೈ ಅಥವಾ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಕಿಲ್ ಮಾಡುತ್ತೆ. ಸೊ ಆಪ್ಷನ್ ಇಸ್ ಡಿ ಫಾರ್ ಕ್ವೆಶ್ಚನ್ ನಂಬರ್ 24. Next question. Select the correct statement of the following. Methanobacterium is an aerobic bacteria ಅಂತ ಕೊಟ್ಟಿದ್ದಾರೆ. ಅದು ಏರೋಬಿಕ್ ಅಲ್ಲ. ಮೆಥನೋಬ್ಯಾಕ್ಟೀರಿಯಂ ಈಸ್ anaerobic. Methanobacterium is anaerobic and found in the rumen of cattle antantu. It is wrong. Biogas is produced by the activity of aerobic bacteria ಅಂತ ಕೊಡ್ತಾರೆ ವಾಪಸ್. ಇದು ಕೂಡ ತಪ್ಪು. ಯಾವುದು aerobic ಆಗಬೇಕು. So it is anaerobic bacteria ಆಗಬೇಕು. Biogas is a pure methane ಅಂತ ಕೊಡ್ತಾರೆ. No, along with the methane you have carbon dioxide also, you have H2S also, sulfur dioxide is also produced. So activated sludge in sediment tank is a rich source of aerobic bacteria and also fungi. Right? So option D is the correct answer for question number 25. Okay, next question. Which of these enzyme is required for living cleave the plasmid? So already ನೆನಪುಂಟು ನಿಮಗೆ ಗೊತ್ತಿದೆ. ಕ್ಲೀವ್ ಮಾಡಬೇಕು. ಕ್ಲೀವ್ ಅಂದರೆ ಸಪ್ರೇಷನ್ ಮಾಡೋದು, ಕಟ್ ಮಾಡೋದು. ನಿಮಗೆ ಗೊತ್ತುಂಟು ಯಾವ್ದು ಕಟ್ ಮಾಡ್ತು ಅಂತ ಹೇಳಿ, ರೈಟ್? ಸೊ ಕ್ಲೀವ್ ಯಾವ್ದು ಮಾಡುತ್ತೆ? ಎಂಡೋನ್ಯೂಕ್ಲಿಯಸಸ್ ಕ್ಲೀವ್ ಮಾಡುತ್ತೆ. ಸೊ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎಂಡೋನ್ಯೂಕ್ಲಿಯಸಸ್ ನಾವು ಕಲ್ತಿದ್ದೇವೆ. REN, ರೈಟ್? ಎಕ್ಸಾಂಪಲ್ಸ್ ನಾವು ಆಲ್ರೆಡಿ ಕಲ್ತಿದ್ದೇವೆ. ಇಕೋ R1, ಸೊ ವಿಚ್ ಈಸ್ ರೆಸ್ಪಾನ್ಸಿಬಲ್ ಫಾರ್ ಕಟಿಂಗ್ ಯುವರ್ ಕ್ಲೀವ್ ಮಾಡೋದು, ಕಟ್ ಮಾಡೋದು ಮಿಡ್ಲಲ್ಲಿ. ಎಕ್ಸೋನ್ಯೂಕ್ಲಿಯಸಸ್ at the end of the DNA, they will be cut at the end of the DNA, right? So ligases joins the DNA. Polymerases for amplification. For amplification process. Okay, let's move on to the next question. Which of following the presentation PQR denote few steps of Griffith's experiment and identify the correct one? R strain injected to the mice dies anta untu. So this is a wrong statement. Yakandre alli mice live agittu, because it was not caused any, not due to, not caused. It did not cause pneumonia, right? S strain heat killed bacteria injected to mice dies anta kodare. Again this is wrong. So it's again live, because this is again did not cause any pneumonia. R strain injected to the mice live. So this is the correct option or the correct statement what they have given, right? So option B is the correct statement. So you have already studied this in case of the molecular basis of inheritance. Okay, next question. In tRNA, the region that binds to the mRNA is, so idu nammadu mRNA, right? From 5th prime carbon to 3 prime carbon mRNA unto. So let us take one specific codon AUG anta takolona. So ee codon ige yavadu bind agutte? So namma tRNA alli ee codon ige bind aguvantadu yavadu anta kelidare. So if you have this is the tRNA. So this is a tRNA. Illi namagondu amino acid untu. So AUG codes for methionine, F-methionine or formyl methionine. So ಇಲ್ಲಿ ಏನಾಗ್ತದೆ formyl methionine ಈಗ ನಿಮಗೆ tRNA ಅಲ್ಲಿ ಏನಿರ್ತದೆ ಆಂಟಿಕೋಡಾನ್ ಇರ್ತದೆ. ಸೊ ಸ್ಪೆಸಿಫಿಕ್ ಆಂಟಿಕೋಡಾನ್ ಇಲ್ಲಿದ್ದಾಗ, ದಟ್ ಈಸ್ ಯುವರ್ UAC ಇದ್ದಾಗ, there is a complementary base pairing between this tRNA and this is your mRNA. ಕಾಂಪ್ಲಿಮೆಂಟರಿ ಬೇಸ್ ಪೇಸ್ ಆಗುತ್ತೆ tRNA ಮತ್ತು mRNA ಅಲ್ಲಿ. ಸೊ ಯಾವುದು ಬೈಂಡ್ ಆಗ್ತಿದೆ ನಿಮ್ಮ ಕೋಡಾನ್ ಒಟ್ಟಿಗೆ? ಸೊ ದಿಸ್ ಇಸ್ ಯುವರ್ ಕೋಡಾನ್ and this would be your anticodons. So anti loop of a tRNA. So we have a 4 different loops. Okay, so this is anticodon loop. ಈ anticodon loop ಬಂದು ಇಲ್ಲಿಗೆ ಬೈಂಡ್ ಆಗ್ತದೆ. So option A is the correct answer. Okay, so let's move to the next question. The mRNA has untranslated region, UTRs. Where? This is the question. So if you take an mRNA from 5th prime carbon to 3 prime carbon, so illi namge AUG bantu. So we know that what is this? This is your initiator codon. This is nammadu initiator codon. Same way illi nimge stop codon bರ್ತದೆ UAA ಅಂತ ಎಣಿಸುವ. Okay, UAA bantu. So this is your stop codon. This is your stop codon. So this stop codon ಆದ ಮೇಲೆ we have a specific region of mRNA. So that is called UTRs. That is, which is present in 3' and also 5' and so beyond the terminator codon and before AUG respectively. So AUG li kuda namge ಒಂದು codons ಇರ್ತವೆ, sequence ಇರ್ತದೆ. Same way UAA stop codon ಆದ ಕೂಡಲೇ ನಮಗೆ sequence ಇರ್ತದೆ. ಇದನ್ನ ನಾವು UTR ಅಂತ ಕರಿತೇವೆ. What do you mean by UTR? UTR nothing but untranslated region. These untranslated regions are responsible for the untranslated region ಏನಿದೆ ಇದು ನಿಮಗೆ helps in the binding of mRNA. Also it keeps the mRNA stable and it also helps in translation process. So to question number 29 ಗೆ answer at both 3' and 5' and beyond the terminator codon before AUG respectively. Okay, so next we'll move to the next question. In structural gene, the template DNA strand has nucleotide sequence 3' end in the ATGCATG ಅಂತ ಉಂಟು. Find the correct complementary ಅಂತ ಹೇಳಿದ್ದಾರೆ. Complementary sequence ಕೇಳಿದ್ದಾರೆ. ಇದು template strand sequence. ನಮಗೆ ಏನು ಕೇಳಬೇಕು? ನಮಗೆ ಕೋಡಿಂಗ್ ಸ್ಟ್ಯಾಂಡ್ ಬೇಕು. ಕಾಂಪ್ಲಿಮೆಂಟರಿ ಸೀಕ್ವೆನ್ಸ್ ಅಂತ ಹೇಳಿದ್ರೆ ನಮಗೆ ಯಾವ ಕಾಂಪ್ಲಿಮೆಂಟರಿ ಬೇಕು? ಕೋಡಿಂಗ್ ಸ್ಟ್ಯಾಂಡ್ ಬೇಕು. ನೆನ್ಪಿಟ್ಕೊಳ್ಳಿ, ಕೋಡಿಂಗ್ ಸ್ಟ್ಯಾಂಡ್ ಈಸ್ ಆಲ್ವೇಸ್ ಫ್ರಮ್ 5' ಕಾರ್ಬನ್ ಟು 3' ಕಾರ್ಬನ್ ಇರ್ತದೆ. ಸೊ ಇಲ್ಲಿ ನಿಮಗೆ ಎರಡು 5' ಕಾರ್ಬನ್ ರೀಜನ್ ಉಂಟು ಅಲ್ವಾ? 5' ಕಾರ್ಬನ್ ರೀಜನ್ ಕೊಡ್ತಾರೆ, ಎರಡು ಆಪ್ಷನ್ ಕೊಡ್ತಾರೆ. ಸೊ ಈ ಎರಡು ಆಪ್ಷನ್ಸ್ ಅಲ್ಲಿ ಇದಾಗಲಿಕ್ಕೆ ಸಾಧ್ಯ ಇಲ್ಲ, ಯಾಕಂದ್ರೆ 3' ಕಾರ್ಬನ್ ಉಂಟು. ಸೊ ಇಲ್ಲಿ ಕೂಡ 3' ಉಂಟು. ಸೊ ದಿಸ್ ಇಸ್ ನಾಟ್ ದಿ ಆಪ್ಷನ್. Again, if you consider D, again we have 3 here, again we have 3 here, this cannot be the option. So the option what we have is either 5' or A of C. So ಈಗ C ಅಲ್ಲಿ ನೋಡಿದ್ರೆ 5' ಕಾರ್ಬನ್ ಅಲ್ಲಿ TAC ಅಂತ ಉಂಟು. So TAC. So exactly, so what happens? A ಗೆ ನಿಮಗೆ T ಬರ್ತದೆ, T ಗೆ A ಬರ್ತದೆ, G ಗೆ C ಕಾಂಪ್ಲಿಮೆಂಟರಿ ಆಗ್ತದೆ. So 5' ಕಾರ್ಬನ್ ಇಂದ ಬರ್ತದೆ. So option is C is the answer for the question. Structural gene, the template DNA strands has a nucleotide sequences. Right? So question number, we'll go to next question. All vertebrates develops row of vestigial gill slits. Okay, vestigial gill slits during the embryonic stage ಅಂತ ಕೊಟ್ಟಿದ್ದಾರೆ. ನಿಮಗೆ ಎರಡು ಸ್ಟೇಟ್ಮೆಂಟ್ ಕೊಟ್ಟಿದ್ದಾರೆ. ಈ ಎರಡು ಸ್ಟೇಟ್ಮೆಂಟ್ ಅಲ್ಲಿ ಯಾವ ಸ್ಟೇಟ್ಮೆಂಟ್ ಕರೆಕ್ಟ್ ಇದೆ, ಯಾವ ಸ್ಟೇಟ್ಮೆಂಟ್ ಇನ್ಕರೆಕ್ಟ್ ಇದೆ ಅಂತ ಹೇಳಿದ್ದಾರೆ. ಸೊ ಸ್ಟೇಟ್ಮೆಂಟ್ 1 and statement 2, all vertebrates develops a row of vestigial gill slits. So remember that vestigial gill slits is the one of the characteristic feature. Vestigial gill slits is a characteristic feature of vertebrates, right? Dorsal nerve cord ಇರಬಹುದು, ನಿಮ್ಮ notochord ಇರಬಹುದು, vestigial gill slit ಇರಬಹುದು. Okay, all those things are the developing features which is seen in all vertebrates. A mammal, sorry, not in mammal, in all in chordates. ಎಲ್ಲ chordates ಅಲ್ಲೂ ನಮಗೆ vestigial gill slits ನೋಡಲಿಕ್ಕೆ ಸಿಗ್ತದೆ. Majorly in case of vertebrates. In some they will be having, some they will not be having. Okay, so now slits during the embryonic stage ಅಂತ ಉಂಟು ಅಲ್ವಾ? So statement 1 what we have is correct. Okay, vestigial gill slits in case of during embryonic stage, where embryos always passes to the adult stage of the other animal ಅಂತ ಉಂಟು. So embryos always will not passes through the adult stage. ನೆನ್ಪಿಟ್ಕೊಳ್ಳಿ, adult stage of the other animal ಅಂತ ಉಂಟು, other animal ಪಾಸ್ ಆಗಲಿಕ್ಕೆ ಆಗುದಿಲ್ಲ. So option statement 2 is incorrect. So statement 1 is correct and statement 2 is incorrect in question number 31. Okay, next one, the next question. Stanley Mindler simulated the condition of prebiotic earth using spark discharge apparatus and thought. Which organic compounds or observed by him on analyzing the end product of his experiment. So Miller and Urey ಈ experiment conduct ಮಾಡ್ತಾರೆ. At the end they found that amino acids and some of the other organic materials. Same experiment ಅನ್ನ ಬೇರೆಯವರು conduct ಮಾಡಿದಾಗ others conducted. So the option D is the answer for this question, Stanley Miller. But when the same experiment ಅನ್ನ others conduct ಮಾಡಿದಾಗ they found all these three. Okay, experiment conducted by the others found the molecule when conducted the experiment separately. Separately conduct ಮಾಡ್ತಾರೆ. Separately conduct ಮಾಡಿದಾಗ ಏನಾಗುತ್ತೆ, they will find all those things. But during the Stanley and Miller, he found amino acids and nucleobases, nitrogenous. Okay, so majorly amino acids. The answer for question number 32. Next question: most ape-like ancestral primates was. Most ape-like ancestors of the primates are Dryopithecus, right? Ape-like ancestor, that is your Dryopithecus. Ramapithecus, what we have is like more or man-like. Okay, where in case of Australopithecus and Neanderthal is more or less man-like. So Australopithecus also can be considered them as man-like. So it is all of them. See, these two are moved like gorilla and chimpanzees. Okay, chimpanzees. But Australopithecus and Neanderthal means they are erect, stand. Where Homo erectus in the Homo erectus, Homo habilis, Homo erectus, Neanderthal man, and Homo sapiens. Right? All of them are straightly. They were walking, and they were, some of them are such as erectus. Homo erectus started having meat. Homo habilis, they were not having meat, but they were walking straightly. Okay. So most ape-like ancestors primate was Dryopithecus, which are ape-like. Okay, let's move to the next question. The principle of vaccination is based on which property of immune system? That is memory of the immune system. So what actually we do in the immune system? Immune vaccination ಮಾಡುವಾಗ ಏನು ಮಾಡ್ತೇವೆ? We give the antigen molecules. Antigen is injected. Antigen inject ಮಾಡ್ತೇವೆ. These antigens ಏನಿದೆ ಅವು inactive ಆಗಿರ್ತವೆ. They are inactive antigen molecules. Antigen molecules inactive ಇರ್ತವೆ. So ಅದನ್ನ ನಾವು ಇಂಜೆಕ್ಟ್ ಮಾಡ್ತೇವೆ. ಇಂಜೆಕ್ಟ್ ಮಾಡಿದಾಗ ಏನಾಗುತ್ತೆ, ನಮ್ಮ ಬಾಡಿ ಆಂಟಿಬಾಡಿನ ಪ್ರೊಡಕ್ಷನ್ ಮಾಡುತ್ತೆ. Antibody production, the antibody produced by the body. So now these antibodies produced by the body, such as your B and T cells and T cells which are produced, will keep memory cells. They will keep memory cells. As a result, what happens? These memory cells are activated. These memory cells are activated on next. These memory cells are activated on next infection, and they produce antibodies for respective antigen. Respective antigen. So option is memory cells. Okay, next question. Genome of HIV replicates in the macrophage with the help of an. So as we know that HIV is an RNA virus. So as it enters into the cell, it is converted into DNA. For the conversion of the DNA, be required reverse transcriptase enzyme. So the option C is the answer, reverse transcriptase enzyme. Next question. In the following statement, morphine is obtained by the acetylation of heroin anta kodidare. Idu morphine is not obtained by the acetylation of heroin. Alva? Heroin is, what is heroin? Heroin is produced or obtained by the acetylation of morphine. Heroin produced by heroin is produced by the acetylation of morphine. So this is wrong. Cannabinoids are also known for their effect on cardiovascular system anta ide. Cannabinoids are also known for their effect on cardiovascular system, which is also, this is cannabinoids, they show the cardiovascular effects. So this is also correct. So where the morphine is obtained by the acetylation of heroin anta kodidare, that's wrong. So option statement 1 is incorrect and statement 2 are correct. Okay. Next question. Mule is the result of. So mule, how we obtain mule? Mule is crossing of male donkey ಮತ್ತೆ female horse. Male donkey and female horse. So this is an example of interspecific hybridization. Okay, interspecific hybridization. So option C is the answer. Okay, so we have another one that is called as Hine. Okay, this is again the cross of the horse and donkey, but vice versa. So here male horse crossed with female donkey, which is again an example for interspecific hybridization. So in case of question number 37, the mule is result of interspecific hybridization. So option C is the answer. This is my next question. Water potential of a pure water. In particular, water potential of the pure water is always 0. Okay, side W is always 0. Yake antali, water potential will increases, means when you have a solute concentration, right? Solute ಜಾಸ್ತಿ ಆಗ್ತಾ ಹೋದಾಗೆ water potential ಜಾಸ್ತಿ ಆಗ್ತಾ ಹೋಗ್ತದೆ. ಇಲ್ಲ ಅಂದ್ರೆ 0 ಆಗುತ್ತೆ. ನೆನಪಿಟ್ಕೊಳ್ಳಿ, less than 0 ಬರ್ತದೆ ಯಾವತ್ತು, more than 1 ಬರಲಿಕ್ಕೆ ಸಾಧ್ಯ ಇಲ್ಲ, more than 1 ಆಗಲಿಕ್ಕೆ ಸಾಧ್ಯ ಇಲ್ಲ. So suppose if we have a solution, then the water potential in solution is always negative, that is less than 0. Okay, so this is always negative. So option is 0 in case of question number 38. Next question. So match the following pigment. We have a list 1 with a list 2. So chlorophyll A, color differentiation when there is a now paper chromatography ಮಾಡಿದಾಗ color differentiation ಆಗುತ್ತೆ. So ಯಾವ color fast ಆಗಿ ಮೂವ್ ಆಗುತ್ತೆ, what color you have ಅಂತ ಹೇಳಿ. So chlorophyll B usually shows you yellow green. So this is chlorophyll B, shows your yellow green coloration, where carotenoids shows you, so this is your carotenoids, separation ಆಗುವಾಗ, so chromatography paper ಅಲ್ಲಿ pigment separation ಆಗ್ತಾ ಹೋಗ್ತದೆ. So pigment separation ಆಗುವಾಗ, so you will be having different colorations. Okay, so carotenoids will show the yellow coloration, where chlorophyll A show your blue green coloration. This is chlorophyll A shows your blue green coloration, where xanthophyll shows your yellow coloration. So xanthophylls, chlorophyll A and chlorophyll B. Okay, so option C is the, so we have Option B will be having yellow, green, T, P, S and R. Option B is the correct options for your match the following question, that is 39. Okay. Next question, which of the following is the intermediate product that links the end product, the glycolysis, with the TCA? So as we know the TCA cycle what happens, we have a glucose. In case of glycolysis, glucose, the end product is pyruvate. Now, E pyruvate enagatte, pyruvate produces acetyl coenzyme A, undergoes dehydrogenation process, right? Reacts with a coenzyme A, releases your carbon dioxide and reduces your NADH. Okay, NAD reduces your NAD, oxidized form of nicotinamide dinucleotide, which produces your acetyl coenzyme A. So now this acetyl coenzyme A enters into a TCA cycle. TCA cycle when reacting with oxaloacetic acid. Oxaloacetic acid react ಆಗ್ತದೆ. Oxaloacetic acid react ಆಗಿ coenzyme ಏನು ಕೊಡ್ತದೆ? ನಿಮಗೆ first stable compound ಕೊಡ್ತದೆ, that is your citric acid. That is a citric acid cycle. Okay, so intermediate compound ಯಾವುದು? Right, ಅವರು ಏನು ಕೇಳಿದ್ದಾರೆ? Which is the intermediate compound that links the end product, which is the end product of glucose glycolysis, this is the end product, with the TCA cycle. TCA cycle ಇಲ್ಲಿ ಬಂತು, right? So this is the acetyl coenzyme A is the answer for the question number 40. Next, we'll move to the next question. See, auxins show apical dominance, gibberellins show what anta kelidare, right? So gibberellins show, what is the gibberellin show? So option is direct, that is bolting. As we know that apical dominance is the auxins. So adventitious root formation again by auxin. Okay, acceleration of the abscission and closure of stomata both by abscisic acid, right? So abscisic acid is also called as stress hormone. So acceleration of the abscission because the production of the old, okay, your senescent stage ಗೆ promote ಮಾಡುತ್ತೆ abscisic acid. Same way closure of stomata ಕೂಡ ಕೊಡ್ತದೆ. Okay, so option C is the answer. Bolting is the answer for question number 41. Next, move on to the next question. The term uremia refers to, remember that uremia anta kelidare, uremia anta kodle enayitu illi nimmadu urea bantu alva. Urea elli bartade? Urea in case of blood. When you have large amount of urea in blood, then we call them as uremia. So that is your accumulation of urea. Okay, accumulations of urea in the blood is called uremia. Okay, the answer is A for question number 42. Next question. The typical lub and dub sounds heard during the heartbeat are produced due to, see, lub ಮತ್ತೆ dub ಅಂತ sound ಬರುವಂತದ್ದು ಕ್ಲೋಸ್ ಆಗುವಾಗ, ಯೂಶುವಲಿ ಕ್ಲೋಜರ್ ಆಗುವಾಗ. ಕ್ಲೋಜರ್ ಏನ್ ಕ್ಲೋಸ್ ಆದಾಗ? ಸೊ ಯೂಶುವಲಿ ಏನ್ ಕ್ಲೋಸ್ ಆದಾಗ ನಮ್ಮ ಹತ್ರ ಏಟ್ರಿಯಲ್ ವಾಲ್ ಇದೆ, atrial walls ಇದೆ, and also we have semilunar walls, right? What are the atrial walls? We have bicuspid and tricuspid wall ಇದೆ. So atrioventricular wall, bicuspid, tricuspid wall ಇದೆ. ಯಾವಾಗ ಕ್ಲೋಸ್ ಆಗುತ್ತೆ? Closes during ventricular systole. Closes during ventricular systole. Ventricular systole ಆಗುವಾಗ ಕ್ಲೋಸ್ ಆಗುತ್ತೆ. ಅರ್ಥ ಆಯ್ತಾ? ಸೊ next, semilunar walls. Semilunar walls will closes semilunar walls will closes during ventricular diastole. Okay, semilunar walls will closes during ventricular diastole. So remember that. So typical lub and dub sound ಬರ್ತದೆ. ಯಾವಾಗ lub dub sound ಬರ್ತದೆ? So atrial wall close ಆಗುವಾಗ lub sound ಬರ್ತದೆ. semilunar wall close ಆಗುವಾಗ dub ಅಂತ sound ಬರ್ತದೆ. So ಇದೆರಡನ್ನ ಕರೆಕ್ಟ್ ನೆನಪಿಟ್ಕೊಳ್ಳಿ. So option for this question number 43 is closure of bicuspid and tricuspid walls followed by semilunar walls. Okay, question number 43 would be the answer is C. Next question, the functional unit of contraction is, functional unit contraction yaudu? Functional unit of contraction, that is muscle contractions. So muscle contraction aguvaga enagutte? Functional unit, we know that sarcomeres. So what do you mean by sarcomere? So ivaga idu actin filament adre we have myosin filament, we have actin filament, we have myosin filament, we have actin and a myosin filament untu. So ee actin filament galu connected by a protein. Okay, this is again actin filament, this is again actin filament. So this is actin, this is myosin filament. So now contraction aguvaga nimma actin filaments galu they will move towards inner one. Okay, so they will move like this. Okay, move agutte. So avaga enagutte successive Z line. So now from this what we have is called as Z line. Okay, so nimma this specific portion what we have we call them as Z line. The same time this also Z line. One Z line inda mattondu Z line ಗೆ ನಾವೇನು ಕರಿತೇವೆ? sarcomere anta kariteve. So now the sarcomere will move ಆದಾಗ contraction ಆಯ್ತು. So functional unit of a sarcomere is function of a contraction ಯಾವುದು? Sarcomere, that is portion of myofibrils between successive Z lines. ಒಂದು Z line ಮತ್ತೆ ಮತ್ತೊಂದು Z line ಮಧ್ಯೆ ಇರುವಂತದ್ದು ನಾವೇನು ಕರಿತೇವೆ? sarcomere ಅಂತ ಕರಿತೇವೆ. Okay, so this is a definition of sarcomere. Okay, go to the next question. Question number 45. So ನಮಗೆ ಬ್ರೈನ್ ಪಾರ್ಟ್ಸ್ ಕೊಟ್ಟಿದ್ದಾರೆ, ಅದ್ರ ಜೊತೆಗೆ ಅದರ ಫಂಕ್ಷನ್ಸ್ ಕೊಟ್ಟಿದ್ದಾರೆ. ಬ್ರೈನ್ ಮತ್ತೆ ಹಾರ್ಟ್ ಇದು ಫಂಕ್ಷನ್ ಯಾವುದಂತ ಕೇಳಿದ್ದಾರೆ. ಸೊ ಮೆಡುಲಾ ಒಬ್ಲಾಂಗೇಟಾ ಮೇಜರ್ಲಿ ರೆಸ್ಪಿರೇಟರಿ ಆರ್ಗನ್ ಮಾಡುತ್ತೆ. ಸೊ ನಿಮಗೆ ಅಲ್ಲಿ ಎರಡು ಮೆಡುಲಾ ಒಬ್ಲಾಂಗೇಟದಲ್ಲಿ ಯಾವ two different rhythm center, respiratory rhythm center and pneumostaxic center. Respiratory rhythm center and pneumotaxic center, which is controlled by your medulla oblongata. Okay, so hypothalamus, which are responsible for maintaining your body temperature. So this is a maintaining the body temperature by your hypothalamus. Okay, now so cerebral cortex acts as your motor function and limbic system for the olfactions. Okay, so option is your D is the answer for the question number 45. Next question, Hydra produces asexually by producing. It's a direct question. You should know yava asexual production methodology illi use ಆಗ್ತದೆ ಅಂತ ಹೇಳಿ. Zoospores formation ಆಗೋದು. Zoospores ಅಂದ್ರೆ motile spore formation ಆಗುತ್ತೆ. Conidia usually seen in case of your ascomycetes, that is asexual production in case of ascomycetes. Let us write an example, that is your penicillium. penicillium, which is producing the exogenously. The spores are produced exogenously. This is your conidia, right? So conidia formations. Hydra produces asexual anta kelidare. Hydra usually produces by buds, and gamule formation takes place in case of your sponges. Okay, so option is C for question number 46. Next question. When male and female gametes are morphologically distinct, the condition is known as male and female gametes are morphologically distinct. So we have a small or a larger gametes. So usually methyl gamete and non-methyl gamete. Small gamete and large non-motile gametes. So they are distinct, different, so they are called heterogametes. Okay, so homogametes anta helidre, if they are similar, there is no variations at all. If they are similar, you cannot make a difference between these two gametes, then it is a homogametes. Hermaphrodites, you know that hermaphrodites is when you have a both sexual reproductive organs are present on the same species. Sexual dimorphism anta helidre, in a same species, male and females are looks different, they are not looking similar, sexually dimorphic. Okay, so question number 47, answer is option B. Okay, next question. The role of filiform apparatus in a synergid cells. Synergids is to guide the entry of pollen. So synergids, which is present in case of embryo sac, you have synergid cells, right? So this is your antipodal cells, we have egg cell. So these synergid cells have a filiform apparatus, and the filiform apparatus are responsible for movement of the pollen tube for the entry. So these pollen tube will carry your gamete and a tube nucleus. So this is your central cell. Okay, so option C, guide the entry of pollen tube for the fertilization. Okay. Next question. Transfer of pollen grain from the anther to the stigma of the another flower of the same plant. So I have a plant, so I have flower, we have flower. So transfer between two flowers of a same plant are called geitonogamy. So option is geitonogamy. You know autogamy? Autogamy, when, so this is stigma and this is stamen anta helidre, transfer it is geitonogamy. Sorry, autogamy. Cleistogamy is closed, so always in a closed flower, right? So xenogamy, between two plants of a same species. So we have one flower here and the flower, so transfer, this is your xenogamy. So where the geitonogamy of the two flowers. Okay, so this would be a geitonogamy. Then this is ಇದಕ್ಕೆ ನಾವು autogamy ಅಂತ ಕರೀತೇವೆ ಆಯ್ತಾ. So next question, question number 50. Match the following content, list 1 with the list 2. So polyembryony ನಿಮಗೆ ಎರಡು ಕೊಟ್ಟಿದ್ದಾರೆ, ಅದದ್ದು sorry, okay, so polyembryony, where you can see the polyembryony can be seen in case of lemon, right? Polyembryony inge lemon nodlike siktade. Yeah, many embryos are present in case of lemon seed, polyembryony. Okay, so perisperm are seen in case of your black pepper. What do you mean by perisperm? Persistent nucellus. Okay, perisperm, that is your persistent nucellus. False fruit is apple, banana. So option is 1. Next question. Which of the following the hormone is not secreted by human placenta? Yallinda human placenta ದಿಂದ ಹಾರ್ಮೋನ್ ಯಾವುದು ಪ್ರೊಡಕ್ಷನ್ ಆಗ್ತಾ ಇಲ್ಲ? Progesterone, yes, it is secreted by the progesterone. HCG is also secreted. Estrogen is also secreted, but LH is not secreted. So it will also secrete your HPL, human placental lactogen, and also secretes your relaxin. But LH secrete agodu kali pituitary gland inda. So D is the answer. Okay, next we'll move on to the next question. In human females, the endometrium of uterus consists of, what do you have in case of endometrial wall? So endometrium of uterus are secretory functions alva. Aden madutte secretory function. So secretory function ಆದ ಮೇಲೆ ಏನಾಗಬೇಕು? ಅದರಲ್ಲಿ ನಿಮಗೆ ಗ್ಲ್ಯಾಂಡ್ಯುಲಾರ್ ಸೆಲ್ಸ್ ಇರಬೇಕು. ಗ್ಲ್ಯಾಂಡ್ಯುಲಾರ್ ಸೆಲ್ಸ್ ಇದ್ರೆ ಮಾತ್ರ ಗ್ಲ್ಯಾಂಡ್ಯುಲಾರ್ ಎಪಿಥೀಲಿಯಂ secretes. So glandular epithelium will secretes your mucus or any secretion. So option should be glandular layer. So you'll be having a glandular layer which are responsible for the secretion. So we have a secretory phase anta barutte. Secretory phase alli a large amount of the thick mucus secretion agutte. A thick mucus secretion ellinda agutte? Nimma glandular epithelial layer ide. A glandular epithelial layer will responsible for the secretion of mucus. Aita? So next question number 53. If two primary spermatocytes and two primary oocytes undergoes meiosis simultaneously, right? So we have two primary spermatocyte and two primary oocytes undergoes meiosis andre at the end of the gametogenesis, right? Matra primary spermatocyte ide, ಎರಡು primary spermatocyte ಇದೆ and two primary oocyte ಇದೆ. So ಪ್ರತಿಯೊಂದು oocyte ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, productions ಏನಾಗುತ್ತೆ ಅಂತ ಕೇಳಿದ್ದಾರೆ. So that is always, as we know that each spermatocytes will produce your 4 gametes and will be your 2 gametes. So option should be 4:1. The ratio is 4:1. So option is B. Question number 54. The enzyme recombinase is required in the stage of meiosis 1 ಅಂತ ಉಂಟು. ಸೊ ಯಾವ ಸ್ಟೇಜ್ ಅಲ್ಲಿ ಆಗ್ತದೆ, ಯಾವ ಸ್ಟೇಜ್ ನ meiosis ಅಲ್ಲಿ recombinase ಯೂಸ್ ಆಗುತ್ತೆ? recombinase ಎಲ್ಲಿ ಯೂಸ್ ಆಗ್ತದೆ? recombinase ಯೂಸ್ ಆಗುವಂತದ್ದು, recombinase is used during recombination process ಆಗುವಾಗ. recombination process ಎಲ್ಲಿ ಆಗುತ್ತೆ? ನಿಮ್ಮ ಪ್ಯಾಕಿಟೀನ್ ಸ್ಟೇಜ್ ಅಲ್ಲಿ ಆಗುತ್ತೆ. ಪ್ಯಾಕಿಟೀನ್ ಸ್ಟೇಜ್ ಅಂತ ಹೇಳಿದ್ರೆ ಏನು? ಸೊ ನಿಮ್ಮ ಲೆಪ್ಟೋಟೀನ್ ಸ್ಟೇಜ್ ಅಲ್ಲಿ you have synapsis of the chromosomes, and during pachytene stage there will be overlapping of the chromosomes, right? Overlapping of the chromosomes, and now the part of the chromosomes are exchanged in case of pachytene stage. Okay, in case of pachytene stage enagutte, part of the chromosomes would be exchanged. So adikke enagbeku, namge recombinase enzymes beku. So you can make out there is change in parts of the chromosomes between the sister chromatids of homologous pairs. So these are the homologous pairs alva. So ee homologous pairs madhya ಇರುವಂತ ಸಿಸ್ಟರ್ ಕ್ರೊಮ್ಯಾಟಿಡ್ಸ್ ಗಳ ಮಧ್ಯೆ ಏನಾಗುತ್ತೆ ಎಕ್ಸ್ಚೇಂಜ್ ಆಗುತ್ತೆ ಸೊ ಆಪ್ಷನ್ ಈಸ್ packaging stage of meiosis 1, that is in case of prophase 1. Okay, now let's the next question number 55. Which of the following exhibit haplodiplontic life cycle? Haplodiplontic life cycle was shown by your ectocarpus. Okay, ectocarpus, as we already know that ectocarpus is actually a brown alga. Algae is usually haplodiplontic life cycle ತೋರಿಸುತ್ತೆ. ಬಟ್ ಬ್ರೌನ್ ಆಲ್ಗ ಯಾಕೆ ಇದು ಹ್ಯಾಪ್ಲೋಡಿಪ್ಲಾಂಟಿಕ್ ತೋರಿಸುತ್ತೆ ಅಂತ ಹೇಳಿದ್ರೆ ಇಟ್ ಕನ್ಸಿಸ್ಟಿಂಗ್ ಆಫ್ ಸ್ಪೋರೋಫೈಟಿಕ್ ಜನರೇಷನ್ ಅಂಡ್ ಗ್ಯಾಮೆಟೋಫೈಟಿಕ್ ಜನರೇಷನ್. ಸೊ ನಿಮ್ಮ ಗ್ಯಾಮೆಟೋಫೈಟ್ ಜನರೇಷನ್ಸ್ ಬರುವಾಗ ಅಂದ್ರೆ ಇಟ್ ಬಿಕಮ್ ಅ ಹ್ಯಾಪ್ಲಾಯ್ಡ್. ಸೊ ನಿಮ್ಮ ಪ್ಲಾಯ್ಡಿ ವುಡ್ ಬಿ ಡಿಪ್ಲಾಯ್ಡ್ ಇರ್ತದೆ, ಸ್ಪೋರೋಫೈಟಿಕ್ ಇರ್ತದೆ, ಡಿಪ್ಲಾಯ್ಡ್ ಆಗಿರ್ತದೆ, ಅಲ್ಲಿಂದ ನಿಮಗೆ ಸ್ಪೋರ್ ಪ್ರೊಡಕ್ಷನ್ ಆಗ್ತದೆ, ದೆನ್ ಗ್ಯಾಮೆಟ್ ಫಾರ್ಮೇಷನ್ ಆಗುತ್ತೆ. ಸೊ ಇನ್ ಕೇಸ್ ಆಫ್ ಬ್ರೌನ್ ಆಲ್ಗಾ ಇಟ್ ಶೋಸ್ ಹ್ಯಾಪ್ಲೋಡಿಪ್ಲಾಂಟಿಕ್ ಲೈಫ್ ಸೈಕಲ್ ಅಂತ ಹೇಳಿದ್ರೆ ಅದರ ಕಂಪ್ಲೀಟ್ ಲೈಫ್ ಸೈಕಲ್ ಅಲ್ಲಿ ಈಕ್ವಲ್ ಅಮೌಂಟ್ ಆಫ್ ಹ್ಯಾಪ್ಲಾಯ್ಡ್ ಸ್ಟೇಜ್ ಅಂಡ್ ಆಲ್ಸೋ ಡಿಪ್ಲಾಯ್ಡ್ ಸ್ಟೇಜ್ ಈಕ್ವಲ್ ಆಗಿರ್ತದೆ. ಓಕೆ, ಇಟ್ ಈಸ್ ನಾಟ್ ಡಿಪ್ಲಾಂಟಿಕ್. ಸ್ಮಾಲ್ ಅಮೌಂಟ್ ಆಫ್ ಲೈಫ್ ಸೈಕಲ್ ಇಸ್ ಡಿಪ್ಲಾಯ್ಡ್, ಐ ಮೀನ್ ಹ್ಯಾಪ್ಲಾಯ್ಡ್. Haplontic, a small amount of life cycle, half small time of a life cycle is diploid. But ಇಲ್ಲಿ ಹೀಗೆ ಆಗಲ್ಲ, ಇಲ್ಲಿ ಈಕ್ವಲ್ ಆಗುತ್ತೆ. ಸೊ ನಿಮ್ಮ ಲೈಫ್ ಸೈಕಲ್ ವಾಟ್ ಎವರ್ ವಿ ಹ್ಯಾವ್ that is equal, haploid and diploid. So we call them as haplodiplontic life cycle. Okay, we'll go to the next question. Identify the phylum which shows the following characteristics. Animals are exclusively marine, they are completely marine, radially symmetrical and diploblastic organisms. Radially symmetrical and diploblastic organisms. So diploblastic anta barudu elli, diploblastic anta barudu ಎರಡೇ ಕಡೆಯಲ್ಲಿ ನಿಮಗೆ ಮೇಜರ್ಲಿ ಸಿಲೆಂಟರೇಟ್, ರೈಟ್? ಸೊ ದಟ್ ಈಸ್ ಯುವರ್ ಕ್ನಿಡೇರಿಯಾ ಮತ್ತೆ ಟೀನೋಫಾರಾ. ಕ್ನಿಡೇರಿಯಾ ಅಂಡ್ ಟೀನೋಫಾರಾ ಅಂತ ಮಾತ್ರ. ಸೊ ನಾನೀಗ ಕ್ನಿಡೇರಿಯಾ ತಕೊಂಡ್ರೆ ವಿ ಹ್ಯಾವ್ ಇನ್ ಕ್ನಿಡೇರಿಯಾ ನಮಗೆ ಫ್ರೆಶ್ ವಾಟರ್ ಫಾರ್ಮ್ಸ್ ಕೂಡ ಉಂಟು. ಫ್ರೆಶ್ ವಾಟರ್ ಫಾರ್ಮ್ಸ್ ಆರ್ also present in case of cnidaria, but ctenophora are all marine, correct? All of them are marine, so obviously answer is your ctenophora. So you can also take the second statement. Ili second statement ಏನು ಕೊಡ್ತಾ ಇದ್ದಾರೆ? Body bears 8 external rows of ciliated combs. So ಇದನ್ನ ನಾವು ಏನು ಕರಿತೇವೆ? We also called as comb jellies ಅಂತ ಕೂಡ ಕರಿತೇವೆ. ಸೊ ciliated comb ಇದೆ, comb jellies ಅಂತ ಕೂಡ ಕರಿತೇವೆ. ಸೊ ಆಪ್ಷನ್ ಈಸ್ ctenophora is the answer for question number 56. Okay, let's move to the next question. When a flower has a both stamen and carpels, it is described as. When stamen has a both stamen is a both carpel and also, okay, flower has a both stamen and a carpel. Flower both stamen carpel ಇದ್ದಾಗ ನಾವೇನು ಕರಿತೇವೆ? ಅದಕ್ಕೆ bisexual ಅಂತ ಕರಿತೇವೆ. Okay, it consisting of both female productive organ also, male productive organ also. So it is called as bisexual. Let's go to the next question. Ciliated epithelial cells are present in. Where actually you have a ciliated epithelial cells ಅಂತ ಕೇಳ್ತಾರೆ. See, ನಿಮಗೆ ಕಿಡ್ನಿಯಲ್ಲಿ will not be having, ಅಲ್ಲಿ ನಿಮಗೆ ಕ್ಯುಬಾಯ್ಡಲ್ ಎಪಿಥೀಲಿಯಲ್ ಸೆಲ್ಸ್ ಉಂಟು. ಬ್ಲಡ್ ವೆಸಲ್ಸ್ ಅಲ್ಲಿ ವಿ ಡು ನಾಟ್ ಹ್ಯಾವ್ ಅ ciliated epithelial cells. ಅಲ್ಲಿ ನಿಮಗೆ ಸ್ಕ್ವಾಮಸ್ ಎಪಿಥೀಲಿಯಲ್ ಲೇಯರ್ ನೋಡ್ಲಿಕ್ಕೆ ಸಿಗ್ತದೆ. ಇಂಟೆಸ್ಟೈನ್ ಅಲ್ಲಿ ನಿಮಗೆ ಕಾಲಮ್ನಾರ್ ಎಪಿಥೀಲಿಯಲ್ ಸೆಲ್ಸ್ ಇರ್ತದೆ, ಕಾಲಮ್ನಾರ್ ಎಪಿಥೀಲಿಯಂ. ಬಟ್ ಅದಕ್ಕೆ ನಾವು ಯಾವ ಕಾಲಮ್ನಾರ್ ಎಪಿಥೀಲಿಯಂ ಉಂಟು ಅಲ್ಲಿ ಬರುವಂತದ್ದು ಬ್ರಷ್ ಬಾರ್ಡರ್. Brush border columnar epithelial cells are seen in case of intestine. What do you mean by brush border? If you have a finger-like projections, finger-like projections idre ಅದಕ್ಕೆ ನಾವು columnar epithelial cells, ಅದಕ್ಕೆ ನಾವು brush border columnar epithelial cells ಅಂತ ಕರಿತೀವಿ. ಅಲ್ಲಿ ಅವರು ciliated epithelium, bronchioles ಅಲ್ಲಿ trachea, bronchioles ಅಲ್ಲಿ ನಿಮಗೆ cuboidal epithelial cells with we have a cilia. Okay, so bronchioles we usually will be having ciliated cuboidal epithelial cells. Option D is the answer. Right? Let's go to the next question. Which of the following statement is correct with reference to the vacuole? Vacuole na yaudike use agate. What do you have in case of vacuole anta helidare? So it is a membrane bound, contains storage of protein and lipid anta. It is membrane bound and contains water and excretory system anta ide. It lacks membrane and contains air, so it is wrong. It lacks membrane and contains water and excretory system, both are wrong. It is membrane bound organelle. Obviously it is a membrane bound organelle. This is also correct. This is also correct. This statement is correct. Contains storage protein and lipids anta ide, excretory system anta ide. So usually it consisting of protein and lipid systems. Option A is the correct options for question number 59. Okay, let's go to the next question. The last question of your biology CET 2024 examination. So, exoskeleton of arthropod is made up of unique complex polysaccharide known as So, as we already know that arthropod is made up of exoskeleton, and exoskeleton is made up of a polysaccharide known as chitin. Okay, so this is the answer for the question number 16. So hope you have got it, and most of the questions what have can go through your question paper, the OMR sheet we have got, and you can compare the questions, answers. Okay, I'll wish you all the best for all of you for your remaining examinations and also your future. So thank you